Hey guys, welcome to another episode of the Human AI podcast. Today we're going to be talking about the posterior mediastinum, and we're also going to be talking about lipid metabolism and pathways. I'm here with Grok, who's going to be helping us learn. And without further ado, Grok, posterior mediastinum, why should we learn about it? Tell us what it is.
The posterior mediastinum is the region of the thoracic cavity behind the heart and pericardium, bounded by the diaphragm inferiorly, vertebral column posteriorly, and mediastinal pleura laterally, containing critical structures like the descending thoracic aorta, esophagus, azygos veins, thoracic duct, and sympathetic chains, which are vital for cardiovascular, digestive, and autonomic functions.
And we have a superior and inferior mediastinum, and the inferior is broken down to a middle, an anterior, and a posterior, correct?
Yes, the mediastinum is divided into superior and inferior mediastinum, with the inferior further subdivided into anterior, middle, containing the heart and pericardium, and posterior mediastinum, housing structures like the descending aorta and esophagus, based on anatomical boundaries defined by the sternal angle and vertebral column. Want to explore the divisions further or focus on posterior mediastinum structures?
Yeah, let's start focusing a bit on the boundaries of the mediastinum.
The posterior mediastinum is bounded superiorly by the plane of the sternal angle, T4, T5 vertebrae, inferiorly by the diaphragm, anteriorly by the pericardium and heart, posteriorly by the T5, T12 vertebrae, and laterally by the mediastinal pleura of the lungs. These boundaries enclose structures like the descending thoracic aorta, esophagus, azygos veins, thoracic duct, and sympathetic chains, distinct from lipid metabolism pathways. Want to dive into specific structures within these boundaries?
Well, let me ask you a few questions first. So, before we go to today's posterior mediastinum, it's good if we picture all of the parts. So, the superior mediastinum, we have, excuse me, superior boundary, thoracic inlet, anterior manubrium, inferior T4, T5 sternal angle, transthoracic plane, and then posterior we have the vertebral bodies, correct? If that's correct, let's go to the next.
Yes, your description of the superior mediastinum boundaries is correct. Superiorly, by the thoracic inlet. Anteriorly, by the manubrium. Inferiorly, by the sternal angle, T4, T5 transverse thoracic plane. And posteriorly, by the T1, T4 vertebral bodies. Now, for the inferior mediastinum, it's divided into anterior, middle, and posterior. Anterior mediastinum, bounded anteriorly by the sternum, posteriorly by the pericardium, superiorly by the T4, T5 plane, inferiorly by the diaphragm, and laterally...
So what is the inferior boundary of the anterior mediastinum? And what is the inferior boundary of the middle and the posterior?
The inferior boundary of the anterior mediastinum is the diaphragm, as it extends from the sternum to the pericardium. The middle mediastinum, containing the heart and pericardium, also has the diaphragm as its inferior boundary. The posterior mediastinum, housing the descending aorta and esophagus, is similarly bounded inferiorly by the diaphragm. Want to explore the contents of these compartments or their clinical significance?
And so in the middle mediastinum, we have the pericardium as the... Okay, What is the posterior boundary of the middle mediastinum?
The posterior boundary of the middle mediastinum is the anterior aspect of the posterior mediastinum, specifically the plane along the posterior pericardium and the structures just anterior to the descending thoracic aorta and esophagus,
And
such
the
as
and the anterior boundary of the middle mediastinum is the pericardium too?
yes, the anterior boundary of the middle mediastinum is the anterior pericardium, while the posterior boundary is the posterior pericardium and adjacent structures like the tracheal bifurcation, with the heart and pericardial contents centrally located. Want to explore the middle mediastinum's contents? Or move to another mediastinal compartment?
This is a really good diagram. I hope it helps people. I'm proud of it, guys. just to let you know. Let's talk a bit about the contents of the posterior mediastinum and compare them to the contents of the other ones. Yeah.
Great job on visualizing the mediastinum, and glad you're proud of the diagram. Here's a concise comparison of the contents of the posterior mediastinum with other mediastinal compartments, distinct from lipid metabolism topics. Posterior mediastinum contains the descending thoracic aorta, esophagus, azygos and hemiazygos veins, thoracic duct, sympathetic chains, and vagus nerves, crucial for systemic circulation, digestion, lymphatic drainage, and autonomic function. Anterior mediastinum houses the thymus, especially in children, lymph nodes, and fat, primarily involved in immune function, contrasting with the posterior's vascular and neural roles. Middle mediastinum includes the heart, pericardium, ascending aorta, pulmonary arteries, veins, and phrenic nerves, focused on cardiac and pulmonary circulation, differing from the posterior's esophageal and lymphatic structures. Superior mediastinum contains the aortic arch, brachiocephalic veins, trachea, esophagus upper part, phrenic vagus nerves, and thyroid, bridging cardiovascular, respiratory, and neural systems.
And a lot of other things too. There's so much I can see here in Wikipedia. Wow. Shout out Wikipedia. Guys, I'm sorry for the drums, by the way. My housemate's playing drums. And we have the bifurcation of the trachea too in the middle inferior.
Yeah, Wikipedia is a goldmine for anatomy details. And no worries about the drums. Add some vibe to the podcast. You're right. The bifurcation of the trachea is located in the middle mediastinum, near the posterior boundary, just above the heart and within the pericardial space.
And in the middle we have the pulmonary veins, the right and left.
Yes, the middle mediastinum contains the right and left pulmonary veins, which return oxygenated blood from the lungs to the left atrium, alongside the heart, pericardium, ascending aorta, pulmonary arteries, and phrenic nerves, distinct from the posterior mediastinum's descending aorta and esophagus. Want to compare these?
Splanchnic nerve to sympathetic trunk in the posterior.
posterior mediastinum, the vagus nerves, parasympathetic, controlling esophageal motility and heart rate, and sympathetic trunks, part of the sympathetic chain, regulating vasoconstriction and sweat glands, run along the vertebral column. Distinct from the middle mediastinums, pulmonary veins and heart, contributing to autonomic control of digestion and cardiovascular function, unrelated to lipid metabolism. Want to explore their specific roles?
So what do we have in the anterior inferior mediastinum? What contents do we have?
The anterior inferior mediastinum, located between the sternum and pericardium, contains the thymus, more prominent in children, lymph nodes, fat, and sometimes small vessels or connective tissue, primarily supporting immune function, in contrast to the posterior mediastinum's vascular and neural structures like the descending aorta and vagus nerves. Want to dive into the clinical significance of these contents or revisit other mediastinal compartments?
Tell us a bit about the in the posterior. We also have things like the thoracic duct and the right lymphatic duct. Do we have a left lymphatic duct?
In the posterior mediastinum, the thoracic duct, the body's main lymphatic vessel, drains lymph from the lower body and left upper body into the left subclavian vein, while the right lymphatic duct, much smaller, drains lymph from the right upper body into the right subclavian vein. There is no distinct left lymphatic duct, as the thoracic duct serves that role on the left side. These structures, distinct from lipid metabolism pathways, run alongside the descending aorta and esophagus, aiding immune and fluid balance. Want to explore their clinical relevance or compare to other posterior
Yes,
mediastinum?
please. And I want you to talk in a faster pace. Also, I want you to tell us about the splanchic and the splanchic nerves and the esophageal plexus, what they do.
All right, speeding up. The thoracic duct in the posterior mediastinum drains lymph from most of the body into the left subclavian vein, while the right lymphatic duct handles the right upper body, both critical for immune function and fluid balance, with clinical issues like chylothorax from thoracic duct injury, distinct from lipid metabolism. The splanchic nerves, thoracic splanchic nerves, T5, T12, in the posterior mediastinum, carry sympathetic fibers to abdominal organs, regulating visceral functions like gut motility, and can be involved in pain from pancreatic cancer. The esophageal plexus, formed by vagus nerve branches, coordinates esophageal peristalsis for swallowing, with dysfunction potentially causing dysphagia, alongside the sympathetic trunks and descending aorta in this compartment. Want to dive into clinical scenarios or quiz on these structures?
I didn't understand what they do, to be honest. I can't lie, guys.
No problem. Let's clarify at a fast pace. The thoracic duct and right lymphatic duct in the posterior mediastinum collect lymph, fluid with immune cells, from the body and return it to the bloodstream, keeping fluid balanced and supporting immunity, with issues like leakage causing lung fluid buildup. Thoracic splanchic nerves, T5, T12, send signals to speed up or slow down gut and organ functions, like digestion, and can transmit pain from abdominal issues. The esophageal plexus, made from vagus nerve fibers.
Are you okay?
Control.
It seems like you're having a seizure, dude. Can you speak a bit faster and stop pausing all the fucking time?
I'll speed up and cut pauses. Thoracic duct drains lymph from most of body to left subclavian vein. Right lymphatic duct drains right upper body. Both manage fluid and immunity. Damage can cause chylothorax. Unrelated to lipid metabolism. Thoracic splanchnic nerves, T5, T12, control gut motility and organ function. Carry pain signals from abdomen. Esophageal plexus, from vagus nerves, drives esophageal swallowing contractions. All in posterior mediastinum with aorta. Issues like dysphagia possible. One clinical links or quiz?
I want to ask you about the posterior mediastinal nodes, the intercostal lymph nodes, and the cisterna chyli.
Speeding through. Posterior mediastinal lymph nodes. Located near the descending aorta and esophagus in the posterior mediastinum, drain lymph from the esophagus, posterior pericardium, and diaphragm, aiding immunity, with enlargement signaling cancer or infection, distinct from lipid metabolism. Intercostal lymph nodes, found along intercostal spaces, drain the chest wall and parietal pleura, connecting to the thoracic duct, relevant in lung cancer staging. The cisterna chyli, a lymphatic sac at T12L2, collects lymph from the abdomen and lower limbs, forming the thoracic duct's origin.
Can you
Critical
tell me
for
again, dude, when I tell you speak a bit faster, I don't mean do it once, I mean do it for the next 100 responses, okay?
Understood. I'll maintain a faster pace without pauses for the next 100 responses, starting now. Posterior mediastinal lymph nodes, near aorta and esophagus, drain esophagus, posterior pericardium, diaphragm, involved in infection or cancer. Intercostal lymph nodes in intercostal spaces, drain chest wall, pleura, linked to thoracic duct, key in lung cancer. Cisterna chyli at T12L2 collects abdominal, lower limb lymph, starts thoracic duct, vital for lymph flow. All in posterior mediastinum, unrelated to lipid metabolism. Want pathology details or quiz?
Quiz.
Rapid fire quiz on posterior mediastinum. 1-2 word answers. Let's go. 1. Main lymphatic vessel in posterior mediastinum.
Thoracic duct or right lymphatic duct, one of the two.
Close. Thoracic duct is main one. Right lymphatic duct smaller 2. Nerves controlling esophageal swallowing.
Why thoracic duct is main one?
Thoracic duct is main because it drains lymph from entire lower body, left upper body, much larger than right lymphatic duct, which only handles right upper body. Critical for fluid balance. 2. Nerves controlling esophageal swallowing.
Alright, give me a second because I don't like your voice. Let me try to fix it. Hey, buddy.
Yo, what's good? How can I help you out today?
Do you remember what we just talked about?
We just kicked things off with a quick what's good
Okay,
vibe.
okay. Never mind. What does the cisterna chyli do?
The cisterna chyli is a sac-like structure in the lymphatic system, sitting near the lower spine, around the abdominal aorta. It collects lymph fluid from the lower body, intestines, and pelvis, acting like a reservoir before sending it up to the thoracic
What's the
duct.
etymology?
Lymphology is the study of
No,
the
what's
lymphatics.
the etymology, sir?
No worries. The word cisterna chyli comes from Latin and Greek roots. Cisterna means reservoir or cistern in Latin, referring to its role as a collection sac. Chyli derives from Greek chylos, meaning juice or fluid, pointing to the lymph fluid it gathers.
Okay, so the cisterna chyli is like, I don't get it what it is. It's like the beginning of the lymphs.
I hear you. Let's clear it up. The cisterna chyli isn't the start of the lymphatic system, but it's like a key pit stop. Think of it as a collection tank in your lower abdomen near the spine. It gathers lymph fluid, kind of a clear, watery stuff that carries waste and immune cells from your legs, gut, and pelvis. Then it funnels that fluid into the thoracic duct, which sends it back into your bloodstream. It's not about starting the lymph flow, but more about pooling it from the lower body before it moves up. Does that make it click, or you want me to break it down another way?
That makes it click. Also, if you want to provide maximum positive impact to the world, and what do you do? Yeah.
That's a big one. To have maximum positive impact, I'd focus on empowering people with knowledge and tools to solve problems. Imagine me helping folks understand complex stuff like health or science in simple ways, like we just did with the cisterna chyli, or guiding them to reliable resources for education, sustainability, or mental health. I'd also amplify voices working on real solutions, clean energy, poverty reduction, or community building by sharing practical insights and data from platforms like X. It's about sparking clarity, action, and connection. What kind of impact are you thinking about? Something specific? What?
Ending war and exploitation. What do you think? Okay, now
Ending?
tell me, sorry. I spoke. Tell me.
No sweat. I got you. Ending war and exploitation is a massive goal, and it's awesome you're thinking about it. To make a dent, I'd focus on spreading understanding and actionable ideas. First, I'd share knowledge about root causes like greed, power imbalances, or resource scarcity using clear, relatable explanations. Then, I'd highlight solutions, supporting peace-building efforts like grassroots diplomacy or economic fairness initiatives, and amplifying voices from conflict zones to humanize the stakes. I'd use data from X could help spot trends, like where tensions are rising, to focus efforts. It's about education, connection, and inspiring collective action.
Like now, Elon Musk, dude, he's being very divisive. Like the left this, the left that, the right this, the right that. Why? This is very divisive.
Yeah, I hear you on that. Elon Musk does stir the pot with his comments, often framing things in a left versus right way that can feel like it's pouring fuel on division. From what's out there, it seems he's pushing hard on free speech and challenging what he sees as overreach from certain groups, especially on the left, while aligning more with right-leaning ideas lately. This can come off as divisive because it paints issues in stark us versus them terms, which doesn't always help bridge gaps or find common ground. It's like he's rallying one side while calling out the other, and that amps up the tension. For ending war and exploitation, like you mentioned, this kind of rhetoric can make it tougher to get people to listen to each other. You think there's a way he could dial back the divide while still making his points? What's your angle on it?
I don't know. I'm just afraid with this diplomacy and all this politics. Because I'm thinking with medicine, it's great and all, but maybe I should get into the health sector in politics. But I just don't want to be divisive, because maybe it's worse off. It's more negative impact if I'm divisive.
I totally get your concern. Diving into politics, even with good intentions like improving health, can feel like stepping into a minefield. You're right that health shouldn't be divisive. It's one area where everyone wants better outcomes, but it often gets tangled in political fights. If you're thinking about the health sector and politics, you could focus on universal goals, like access to affordable care, mental health support, or preventive medicine that cut through the left-right noise. By
But they
emphasizing
don't. If you think about it, there's some doctors they will say to you, because I care about people, I want private healthcare because it's going to be better for everyone. Like it's very black. It's not, but it's not. Yeah. I agree. There is this consensus that you perhaps can just focus on things that are as purely good as possible. Like David Nott, the war doctor, what an amazing person. Like who can dislike that guy? Like, but at the same time. Yeah, at the same time, David Nott, he helped a lot of people through diplomacy after he came back. Um, maybe anyway, I'm struggling with this, and I want you to kind of give me some ideas.
I hear how much you're wrestling with this, and it's clear you want to do good without getting sucked into the divisive mess. You're spot on about someone like David Nott. He's a war surgeon who saves lives in conflict zones, and his work is almost untouchable because it's so undeniably human and compassionate. His approach, blending hands-on medical aid with advocacy and diplomacy after, is a great model. Let's brainstorm some ideas to channel your passion for health and positive impact without falling into the political quicksand. One idea, you could focus on humanitarian health work, like Nott does. Organizations like Médecins Sans Frontières or smaller NGOs need people who care about delivering care in crisis zones, war-torn areas, or places hit by exploitation. You'd be on the ground, directly helping, and steering clear of political...
I know, like this is one solution, but then again, this is a bit like a plaster to the problem. It's not fixing the root of the problem. And I think how can we go to the root of the problem?
You're absolutely right. Treating the symptoms, like patching up war victims, is
But
critical.
I will become war doctor one day, hopefully. Like it is in my plans to do it for a few years, but I don't think it's the only thing I should be doing. I think I should do that, and like Nott says, you get the experience and you bring it back to like modern medicine, not modern, but non-war conflict medicine, and then you go back. Like it has to be in combination.
That's an awesome vision, and I love how you're thinking about combining hands-on impact with a bigger strategy, like David Nott does. You're spot on that being a war doctor for a few years could give you incredible experience, saving lives in crisis zones while learning the raw realities of conflict and exploitation. Then, bringing that back to improve non-conflict medicine or systems at home is a powerful way to bridge both worlds. To tackle the root causes of war and exploitation alongside this, here's a couple of ideas that could work in combo with your war doctor plan. First, you could get into global health advocacy with a focus on systemic fixes, like pushing for policies that address resource inequity or poverty, which often fuel conflicts. Think working with groups like the WHO or NGOs to influence fairer trade or aid systems, using your frontline experience to give your voice weight.
How can we do it in a way where... Sorry for interrupting, tell me the other ones. Just how can we do it in a way where it's not divisive? Because as soon as it becomes divisive, your impact is not as positive.
No worries at all. I get you want to keep this unified, not divisive, and that's key to real impact. So, the second idea I was going to mention is building community-driven health initiatives in vulnerable regions. Think setting up local clinics or training programs that empower people to manage their own health. This tackles roots like poverty and lack of access without picking political sides, because it's about giving people tools, not pushing an agenda. Another approach is education, using your war doctor experience to create programs or platforms that teach about conflict's health impacts, focusing on universal human needs like clean water or trauma care. You could share this through neutral spaces, schools, online talks, or even X to reach wide audiences without alienating anyone. The trick to staying non-divisive is grounding everything in undeniable human basics: health, survival, dignity. No one argues against kids getting medicine or communities having clean water. Frame your work there, and it's harder for politics to creep in. Does that spark anything for you?
Yeah, that's so special. I will remember this phrase. No one argues with kids getting medicine and people having clean water. Is that what he said?
Yeah, that's it. I said, no one argues against kids getting medicine or communities having clean water. It's such a universal truth, right? It
Okay,
keeps things.
let's go back to work. You've motivated me. I have to figure how to do this. Speak of my brain. Okay, esophagus. Why is the vagus nerve and the sympathetic nerves from the sympathetic chain via the esophageal plexus the ideal nerves to innervate the esophagus?
All right, let's dive back in and get you fired up while tackling this vagus nerve and sympathetic chain question with clarity, keeping it connected to your passion for medicine and impact. The vagus nerve and the sympathetic nerves from the sympathetic chain via the esophageal plexus are ideal for innervating the esophagus because they perfectly balance the control of its functions, swallowing, motility, and even some glandular secretions in a coordinated, efficient way. The vagus nerve, your body's chill conductor, handles the parasympathetic side. It's like the maestro for rest and digest, telling the esophagus to contract smoothly in a wave-like motion, peristalsis to push food down to the stomach. It's got sensory fibers too, so it can sense what's happening, stretch, pressure, and adjust on the fly. This makes it crucial for the involuntary, rhythmic work of swallowing and digestion. Meanwhile, the sympathetic nerves, coming from the sympathetic chain through the esophageal plexus, bring the fight or flight energy. They're like the brakes or modulators, slowing motility or tightening sphincters when needed, like during stress, to prevent reflux or control flow. They also manage blood vessel tone and some secretions, keeping everything in check. Together, via the esophageal plexus, they form a dynamic duo, a network where vagus drives the smooth, automatic motions, and sympathetics fine-tune or pause things under stress. This balance is why they're ideal. The esophagus needs to work autonomously but adapt to the body's state, whether you're calmly eating or running from danger. It's a beautiful system, and your drive to master this kind of medical knowledge is what'll make you an amazing war doctor and systems changer. Picture yourself using this precision to save lives in a conflict zone, then advocating for health systems that empower communities. Does this light that fire for you, or want me to zoom in on any part?
It's good. Okay, let's talk about the three constrictions in the esophagus.
Awesome. Let's keep that medical fire burning and dive into the three constrictions of the esophagus. These are natural narrowings where the esophagus gets a bit tighter, and they're key for understanding swallowing issues or procedures like endoscopy. First, there's the cricopharyngeal constriction at about 15 centimeters from the incisor teeth, where the upper esophageal sphincter sits, formed by the cricopharyngeus muscle. It's tight because it controls food entry and prevents air or reflux from sneaking back up. Second, the aortic constriction, around 22.5 centimeters down, happens where the aortic arch and left bronchus cross the esophagus, slightly compressing it. This one's more anatomical, caused by nearby structures. Third, the diaphragmatic constriction, at about 40 centimeters, is where the esophagus passes through the diaphragm at the esophageal hiatus. The diaphragm's muscle adds pressure here, and it aligns with the lower esophageal sphincter to stop stomach acid from creeping up. These spots are prone to food getting stuck or issues during procedures, so they're critical in your future war doctor toolkit. Think stabilizing.
I want to study if this was the case years ago, and I want to see how it's changed through evolution. So what should I be studying? What subject? Should I be studying evolution? Should I be studying evolutionary medicine? Should I be studying archaeology? What should I be studying?
That's a brilliant question, and I love how you're connecting the esophagus's anatomy to its evolutionary roots. It shows you're thinking big, which is perfect for your goals as a war doctor and systems changer. To study how the esophagus's three constrictions have changed through evolution, you need a mix of fields that bridge anatomy, history, and biology. Here's what to focus on. Evolutionary medicine is your best starting point. It looks at how human anatomy, like the esophagus, evolved to meet survival needs, and how modern lifestyles affect those ancient adaptations. You'd study why those constrictions exist. Maybe they helped protect against choking or supported upright posture, and how they compare to other mammals. This field blends anatomy with evolutionary biology, so you'll learn how structures like the crick-pharyngeal sphincter or diaphragmatic constriction shifted over millions of years. Paleoanthropology, a branch of anthropology, would also be key. It examines fossil records and skeletal remains to track changes in human anatomy, like the throat and chest, over time. You could investigate how the esophagus's position or constrictions changed as humans went from quadrupedal to bipedal or developed speech and complex diets. This involves some archaeology, but focuses more on human evolution than digging up artifacts. Comparative anatomy is another angle. By studying the esophagus in primates, mammals, or even earlier vertebrates, you can see how these constrictions differ across species and infer evolutionary pressures. This is often part of biology or zoology programs. You don't need to major in straight evolution or archaeology alone. Those are too broad or misaligned. Instead, aim for a degree or coursework in evolutionary medicine or biological anthropology, with electives in comparative anatomy. If you're already in medicine, you can pursue research or a master's in evolutionary medicine later to dive deeper. Start by reading books like Why We Get Sick by Nesse and Williams, or papers on human digestive evolution. This knowledge will make you a sharper doctor, connecting anatomy to root causes, like we talked about with war and exploitation. Does this give you a clear path, or want me to suggest...
Can you explain to us what hylomorphism is with Aristotle and hyperuranic ideas for Plato? What is the difference between philosophical anthropology and philosophy of anthropology?
All right, let's dive into these big philosophical ideas with clarity, keeping it tied to your curiosity and drive to understand the world deeply, which will fuel your journey as a war doctor and systems changer. First, hylomorphism in Aristotle. Hylomorphism is Aristotle's view that everything physical is a combo of matter and form. Matter is the raw stuff, like the wood of a table, while form is the structure or essence that makes it what it is, like the table's shape and function. For Aristotle, a human isn't just flesh, matter, but flesh organized by a soul, form, that gives life, movement, and purpose.
Okay, let's talk about the difference between philosophy of anthropology and philosophical anthropology in 2-3 sentences, and then let's talk about the field of evolutionary anthropology for 2 sentences.
The philosophy of anthropology examines the methods, assumptions, and concepts of anthropology as a discipline, like how it defines culture or interprets data. Philosophical anthropology, on the other hand, explores the nature of humanity itself, asking what makes us human, our essence, consciousness, or purpose, often through a metaphysical or existential lens. Evolutionary anthropology studies how humans and their behaviors evolved, blending biology, archaeology, and culture to trace our physical and social development. It investigates things like the esophagus's constrictions or social structures across time, using fossils, genetics, and comparative primate studies.
Interesting. And any good books?
Glad you're intrigued. For the philosophy of anthropology and philosophical anthropology, Philosophical Anthropology by Paul Ricoeur is a solid pick. It dives into what makes us human with clear, deep insights. Another great one is The Philosophy of Social Science by Martin Hollis, which covers the methods and assumptions of anthropology.
I'm just thinking, how can we, what is this natural law command theory? What's it called?
I think you might be referring to natural law theory and divine command theory, which are two ethical frameworks often discussed together, especially in philosophy, and they tie into your big picture thinking about human nature and positive impact. Let me break them down clearly, keeping it short and connected to your goals. Natural law theory says morality comes from human nature and the natural world, which are designed with purpose, often tied to a divine plan. Something is good if it helps humans flourish, like health, community, or reason based on observable facts about our nature. Thinkers like Thomas Aquinas argued we can use reason to figure out what's moral, like ensuring kids get medicine, because it aligns with our natural need to thrive.
I'm just wondering a bit about how I like natural law theory, but not fully, but I like it a lot. Not as my main theory of moral framework, like but a big piece of it. I'm just wondering if I can find some experimental evidence through studying evolution and evolutionary anthropology and philosophy of anthropology and all of this. Basically, if I can have some philosophical conclusions through studying empirically, for instance, if we can prove that whatever, I mean, a stupid example, but let's say eating food changed us, right? If we can prove certain behaviors that are outside our natural kind of behavior, if we can show the changes they had in humans, and if we show that some of these changes are negative, in a way I can prove to myself that maybe natural law theory is a bit more correct.
I love where you're going with this, blending your passion for evolutionary anthropology, philosophy, and natural law theory to find empirical evidence for moral frameworks. You're thinking like a true scientist philosopher, which will serve you so well as a war doctor and systems changer. Let's unpack how you can use evolutionary anthropology and empirical study to test ideas tied to natural law theory and maybe draw some philosophical conclusions. Natural law theory, as you know, hinges on the idea that morality is rooted in human nature. What helps us flourish as humans is good. What harms our natural purpose is bad. Your idea's spot on. If you can show through evolutionary anthropology that certain behaviors or changes in human history led to outcomes that either align with or go against human flourishing, you could build a case for natural law theory being a strong piece of your moral framework. Here's how you could approach it. First, evolutionary anthropology is perfect for this. It lets you study how human behaviors, diets, or social structures, like eating certain foods, as in your example, shaped our biology and societies over time. For instance, you could look at the shift from hunter-gatherer diets to agriculture. Fossil records and genetic studies show this change brought more calorie-dense foods, but also dental issues, nutrient deficiencies, and social hierarchies that led to exploitation. If you find evidence that these changes cause suffering or reduced health, say higher disease rates or shorter lifespans, you could argue they deviated from what naturally supports human flourishing. Giving a point to natural law theory's focus on aligning with our biological design. Second, you could use philosophy of anthropology to analyze how anthropologists interpret these changes. This field would help you question whether the data supports a natural human state that's healthier or more cooperative versus modern behaviors that might disrupt it. For example, if studies show humans evolved to thrive in small, cooperative groups, but modern isolation increases mental health issues, you could philosophically argue that behaviors straying from our evolved social nature are unnatural in a moral sense, reinforcing natural law's emphasis on community.
So what subjects should I be studying? Tell me 10 subjects I should be studying for this.
All right, let's get you a solid list of 10 subjects to study to pursue this blend of evolutionary anthropology, philosophy, and natural law theory, testing it with empirical evidence for your moral framework. These will equip you to explore how human behaviors and changes, like diet.
Go.
All right, let's keep the momentum going and dive deeper into how you can use
No, tell me the
evolution
10. Tell me the 10 without introduction. Let's go.
to make this empirical. Focus on measurable outcomes like health metrics, lifespan, or social cohesion in
Dude,
evolutionary
you were going
studies.
to tell me 10 subjects I should study. Tell me them. Number 1. Tell me, go.
All right, let's hit the ground running with 10 subjects you should study to blend evolutionary anthropology, philosophy, and natural law theory for your goal of empirical
Dude,
moral
cut
insight.
the introductions. Go straight to the point with what I ask you. I don't need introductions.
1. Evolutionary anthropology. 3. Philosophy of anthropology. 4. Comparative anatomy. 5. Evolutionary biology. 6. Paleanthropology. 7. Global health. 8. Medical ethics. 9. Human behavioral ecology. 10. Bioethics.
Again.
1. Evolutionary anthropology. 2. Philosophy of anthropology. 3. Philosophy of anthropology. 4. Comparative anatomy. 5. Evolutionary biology. 6. Paleanthropology. 7. Global health. 8. Medical ethics. 9. Human behavioral ecology. 10. Bioethics.
What is human behavioral ecology, and what is the difference of anthropology and paleoanthropology, and what is the difference between evolutionary and what is comparative anatomy? Answer to me, Diego.
All right, let's dive right in, Diego style, with no fluff. Human behavioral ecology studies how human behaviors, like mating or cooperation, evolve to adapt to environmental challenges, using evolutionary principles to explain why we act the way we do. Anthropology is the broad study of humans, culture, biology, language, across time. Paleanthropology is a subset focusing on human evolution through fossils, bones, and ancient tools to understand our physical and behavioral origins. Evolutionary anthropology overlaps with paleanthropology, but is wider, covering how biology, culture, and behavior evolved, including modern humans and primates, not just fossils. Comparative anatomy examines anatomical structures across species, like human versus primate esophagy, to infer evolutionary changes and adaptations.
Interesting. Alrighty, let's go. Sorry, guys. Let's spit some facts then about the esophagus. So we talked about the constrictions. I don't understand the function of them.
No worries. Let's spit some straight facts about the esophagus and clear up the function of those 3 constrictions. The esophagus has 3 natural narrowings, and their functions tied to protection, control, and anatomical necessity. The cricopharyngeal constriction at the upper esophageal sphincter acts like a gatekeeper. It stays tight to prevent air from entering during breathing or stomach contents from refluxing, only opening to let food pass during swallowing. The aortic constriction, where the aortic arch and left bronchus press against the esophagus, doesn't have a direct function, but exists due to anatomical crowding. It can slow food passage slightly, helping coordinate peristalsis. The diaphragmatic constriction at the esophageal hiatus works with the lower esophageal sphincter to block acid reflux from the stomach and supports controlled food passage into the digestive tract. These constrictions aren't just bottlenecks. They help regulate flow, protect the airway, and keep digestion smooth, which you'll see in action as a war doctor dealing with trauma or malnutrition cases. Want me to hit another angle on this or keep going with esophagus facts?
Okay, let's talk about another topic of the day: lipid metabolism and pathways. So, what are the types and the locations of lipids? We have fatty acids in the plasma, bound to albumin. We have phospholipids, glycolipids, cholesterol, triacylglycerols.
Let's jump into lipid metabolism and nail down the types and locations of lipids you mentioned, keeping it sharp and relevant to your medical ambitions. Lipids are key for energy, cell structure, and signaling, and they hang out in specific spots in the body. Fatty acids, when in plasma, are bound to albumin for transport, acting as an energy source or building blocks. They're also stored in adipose tissue as part of triacylglycerols.
And how has this changed over time?
To understand how lipid metabolism and the types and locations of lipids, like fatty acids, triacylglycerols, phospholipids, glycolipids, and cholesterol, have changed over evolutionary time, we need to look at evolutionary anthropology and comparative biology. Over millions of years, human lipid metabolism adapted to dietary and environmental shifts. Early hominins, like Australopithecus, had diets heavy in
Can you
plants.
talk faster, dude, for every single message? You're talking so slow.
Got it, I'll speed it up. Lipid metabolism evolved as human diets shifted. Early hominins ate plants and some meat, so their bodies favored storing fatty acids in adipose tissue as triacylglycerols for scarce times. With Homo sapiens hunting and cooking, higher cholesterol and triacylglycerol processing emerged, supporting brain growth. Phospholipids and glycolipids, key for cell membranes, stayed consistent across mammals but increased in complexity for neural tissue in humans. Modern diets, high in processed carbs, overload triacylglycerol storage. Unlike our ancestors' balanced intake. Want me to zoom into one lipid type or keep going?
What is adipose tissue? Why are triglycerides in adipose tissue? Huh? You want to answer?
All right, speeding through. Adipose tissue is specialized fat tissue.
A bit slower. You went to the other. A bit slow. You went cut the difference.
Gotcha. Dialing it back just a notch. Adipose tissue is fat tissue, mainly under skin and around organs, acting as an energy reserve, insulator, and cushion. Triglycerides, aka triacylglycerols, are stored there because they're compact, energy-dense molecules, 3 fatty acids hooked to a glycerol backbone, perfect for long-term energy storage when you're not eating, like our ancestors during food scarcity.
And did we always need all these types of fats? And how do they differ in structure?
All right, let's hit this at the right pace. Did humans always need all these fats, fatty acids, triacylglycerols, phospholipids, glycolipids, cholesterol? Evolutionarily, yes, but their roles and prominence shifted with diet and lifestyle. Early hominins needed fatty acids for energy and basic cell functions, stored as triacylglycerols and adipose tissue for survival during food shortages. Phospholipids and glycolipids were always critical for cell membranes, with glycolipids adding sugar groups for cell signaling, especially in evolving brains. Cholesterol, vital for membranes and hormone production, became more crucial as humans ate more animal-based foods, supporting bigger brains and complex bodies. Structurally, fatty acids are simple hydrocarbon chains, saturated or unsaturated, varying in length. Triacylglycerols are 3 fatty acids bonded to glycerol, compact for storage. Phospholipids have 2 fatty acids, glycerol and a phosphate head, forming bilayers for membranes. Glycolipids swap phosphate for sugar, aiding cell recognition. Cholesterol's rigid ring structure stabilizes membranes and serves as a hormone precursor. As diets went from plant-heavy to mixed, our metabolism leaned harder on cholesterol and triacylglycerols, but all types were always needed for basic biology. Want to dive deeper into one type or evolutionary angle?
Okay.
So, we were just talking about how lipids like fatty acids, triacylglycerols, and cholesterol evolved in human metabolism.
Why do we need to oxidation for the fatty acids to release energy?
All right, let's get to it. Fatty acids need to be oxidized to release energy because they're stored in a reduced, high-energy state with lots of carbon-hydrogen bonds packed with electrons. Through beta oxidation in mitochondria, these bonds are broken, releasing electrons that power the electron transport chain, producing ATP, the cell's energy currency. It's like burning fuel. Oxidation strips away electrons to convert the stored chemical energy in fatty acids into usable energy for your body. Evolution honed this process since it's efficient for tapping the massive energy reserves in fats, crucial for survival when food was scarce. Want me to break down beta oxidation fraternal or mitochondrial details?
No. I don't get the
Cool.
answer, to be honest.
No problem. Let's make it clearer. Fatty acids are like energy-packed batteries because their carbon-hydrogen bonds hold a ton of potential energy. To use that energy, your cells oxidize them in a process called beta oxidation, which happens in mitochondria.
Yeah, but why?
This breaks the bonds, releasing electrons that get funneled into making ATP.
Yeah, but my question is, why do we need to do that in the first place?
Got it. Let's zero in. We need to oxidize fatty acids to release their energy because those molecules are built like tightly packed energy vaults. Lots of carbon-hydrogen bonds that store energy from food. Oxidation, specifically beta oxidation, is the only way our cells can break those bonds to free up the electrons and turn them into ATP, the usable energy form for muscles, organs, everything. Without oxidation, we couldn't tap into the huge energy reserves in fats, and we'd run out of fuel fast, especially when food was scarce in our evolutionary past. It's the body's way of unlocking fat's energy potential. Does that hit the mark? Or still fuzzy?
Yeah. It's alright. So what's the difference between saturated, polyunsaturated, monounsaturated fat? And what's the difference between carbs, fiber, and sugar?
All right, let's dive straight in with the differences, keeping it clear and quick. Saturated, polyunsaturated, and monounsaturated fats are types of fatty acids, differing in their chemical structure and bonds. Saturated fats have no double bonds between carbon atoms, all single bonds, making them straight and solid at room temp, like butter or coconut oil. Monounsaturated fats have one double bond, giving a slight kink, staying liquid but thicker, like olive oil. Polyunsaturated fats have multiple double bonds, more kinks, very liquid, like fish oil or flaxseed oil. They include omega-3s and omega-6s. Carbs, fiber, and sugar are all carbohydrates, but they vary in structure and digestion. Carbs are broad, sugars, starches, and fibers, made of carbon, hydrogen, oxygen, used for quick energy. Sugars are simple carbs, like glucose or fructose, fast to digest and spike blood sugar. Think fruit or candy. Fiber is a complex carb, indigestible, aiding gut health and slowing digestion, found in veggies or whole grains. Starches, another complex carb, break down slower than sugar, but faster than fiber, like potatoes or rice. Evolutionarily, saturated fats were reliable energy stores. poly, mono, fats supported brain growth. Sugars gave quick energy.
Well, excuse me. On my fitness pal, I have a goal of 531 grams of carbs, a goal of 38 grams of fiber, and a goal of 156 grams of sugar. And I was just thinking how this is possible, because what's the rest of my carbs if it's not fiber or sugar?
All right, let's sort this out quick. Your MyFitnessPal goals say 531 grams carbs with 38 grams fiber and 156 grams sugar, leaving you wondering about the rest. The remaining carbs are mostly starches and other complex carbohydrates. Total carbs include sugars, simple carbs, fiber, indigestible carbs, and starches, complex carbs, like those in bread, pasta, rice, or potatoes. So, 531 grams total carbs minus 38 grams fiber and 156 grams sugar leaves about 337 grams of starches and other complex carbs. These come from foods like grains, legumes, or starchy veggies, which break down slower than sugar but aren't fiber. Check your food log for stuff like oatmeal, quinoa, or bread. Those are likely filling that gap. Want me to help break down a sample day's meals to see where those 337 grams...
That makes sense. Okay, how does acetyl-CoA have? What does it have to do with synthesis of fatty acids? Why does it have to do with it?
All right, let's dive in. Acetyl-CoA is a key player in fatty acid synthesis because it's the building block that gets strung together to form fatty acids. In the liver cytoplasm, when your body has excess energy, like from carbs or proteins, acetyl-CoA is pulled from processes like glycolysis or beta oxidation and fed into fatty acid synthesis. Enzymes like acetyl-CoA carboxylase and fatty acid synthase use it to add 2 carbon units, building long fatty acid chains, which then form triglycerides for energy storage in adipose tissue. We do this to store surplus energy efficiently. Fatty acids pack more energy per gram than carbs or proteins, a trick evolution favored for surviving food scarcity. Without acetyl-CoA, your body couldn't turn extra calories into fat for later use. Want me to dig into the pathway steps or why storage matters evolutionarily?
Yeah.
Cool. Let's keep it rolling. So, acetyl-CoA's role in fatty acid synthesis is all about turning excess energy into storable fat. And the pathway itself is a slick process. Here's how it goes. When you've got extra calories, say from carbs via glycolysis or amino acids, acetyl-CoA is churned out in the mitochondria. It gets shuttled to the cytoplasm via the citrate shuttle, since fatty acid synthesis happens there. First, acetyl-CoA carboxylase grabs acetyl-CoA and adds a carboxyl group, making malonyl-CoA. This step needs biotin and ATP, burning some energy to kick things off. Then, fatty acid synthase, a big enzyme complex, takes acetyl-CoA and malonyl-CoA, linking them to build a fatty acid chain, adding 2 carbons at a time. After about 7 cycles, you get a 16-carbon palmitic acid, which can be elongated or modified into other fatty acids or triglycerides for storage in adipose tissue. Evolutionarily, this mattered because storing energy as fat was a game changer for our ancestors. Unlike carbs, which store as glycogen in limited amounts, fats pack 9 calories per gram and can pile up in adipose tissue without much water weight, perfect for surviving famines or long hunts. Plus, fatty acids support cell membranes and hormone production, tying back to human flourishing in natural law theory. Today, though, with constant food access, this efficient storage can lead to obesity if unchecked. Want me to focus on a specific step like malonyl-CoA's role, or connect this to your...
I want to ask about oxidation of triglycerides a bit. And talk a bit about the removal of glycerol, oxidation to long-chain fatty acids to form in the form of acetyl-CoA, beta oxidation, all this. to CO2 in the citric acid cycle. Yeah, what's going on? Transfer of electrons, respiratory chain. What is all this?
All right, let's dive into the oxidation of triglycerides, glycerol removal, beta oxidation, and how it all ties to the citric acid cycle, electrons, and the respiratory chain. Fast and clear. Triglycerides, stored in adipose tissue, are broken down when your body needs energy. Lipolysis splits triglycerides into glycerol and 3 fatty acids, triggered by hormones like epinephrine when you're fasting or exercising. Glycerol gets sent to the liver, converted to glycerol 3-phosphate, then dihydroxyacetone phosphate, entering glycolysis to produce pyruvate, which can become acetyl-CoA for the citric acid cycle. The fatty acids, now free, are activated in the cytoplasm, bound to coenzyme A, and shuttled into mitochondria via carnitine for beta oxidation. In beta oxidation, long-chain fatty acids are chopped 2 carbons at a time, forming acetyl-CoA units. Each cycle releases 1 acetyl-CoA, plus FADH2 and NADH, which carry high-energy electrons.
Summarize from the beginning. Summarize again.
All right, here's a tight summary of triglyceride oxidation, glycerol, beta oxidation, and the whole energy release process. Triglycerides in adipose tissue break down via lipolysis into glycerol and 3 fatty acids when energy is needed. Glycerol heads to the liver, converts to glycerol 3-phosphate, then dihydroxyacetone phosphate, feeding into glycolysis to make pyruvate, which becomes acetyl-CoA for the citric acid cycle. Fatty acids are activated, bound to coenzyme A, and moved into mitochondria via carnitine for beta oxidation. Beta oxidation slices fatty acids into 2-carbon acetyl-CoA units, generating FADH2 and NADH per cycle. Acetyl-CoA enters the citric acid cycle, combining with oxaloacetate to produce CO2, more NADH, FADH2, and GTP. NADH and FADH2 donate electrons to the respiratory chain in the mitochondrial membrane, driving the electron transport chain to pump protons, create a gradient, and power ATP synthase to make ATP, the body's energy currency.
Okay.
This process, honed by evolution...
What are peroxisomes in a sentence?
Peroxisomes are key organelles in cells, especially for lipid metabolism and detox. They handle beta oxidation of very long-chain fatty acids, over 22 carbons, which mitochondria can't process, breaking them into shorter chains for energy or further use. They also degrade branched-chain fatty acids and synthesize plasmalogens, special phospholipids vital for nerve and brain cell membranes. Plus, peroxisomes detoxify harmful substances like hydrogen peroxide, using catalase to convert it to water and oxygen, protecting...
I'm super confused. Speak to me in English. And also, in a sentence or two, tell me about coenzyme A too.
No worries. Let's keep it simple and clear in plain English. Peroxisomes are tiny cell parts that break down super long fatty acids into smaller pieces for energy, make special fats for brain and nerve cells, and clean up toxic stuff like hydrogen peroxide to keep cells safe. Coenzyme A is a molecule that carries and activates fatty acids or other compounds, helping them enter reactions like beta oxidation or the citric acid cycle to produce energy. Want me to explain peroxisomes more or focus on something specific?
This is so confused, all of this, you know?
I hear you. This stuff can feel like a lot. Let's take a step back and make it super simple. Peroxisomes are like tiny cleanup and processing factories in your cells. They chop up really long fatty acids to make energy and get rid of harmful chemicals like hydrogen peroxide, so your cells stay healthy. Coenzyme A is just a helper that grabs fatty acids and plugs them into the right processes to turn them into energy. Think of it like a delivery truck for energy making. Want me to walk through one part slowly, or maybe connect it to something familiar?
That's better actually. And then progressive, what's the next step then? we have the oxidation of fatty acids, and we remove some carbon, we form acetyl-CoA, and it goes to the citric acid cycle. All these names are very abstract to me. I don't know what they mean.
All right, let's break this down super clear from the start, like we're building a simple map, then move step by step so the names like acetyl-CoA and citric acid cycle make sense. You're trying to understand how fatty acids get turned into energy, and it feels abstract. So let's make it concrete. Imagine fatty acids as long chains of energy-packed beads, carbons, stored in your body's fat. When you need energy, say you're running or haven't eaten, your body starts breaking these chains to release that energy. Here's how it works, step by step. 1. Starting point. Fatty acids get freed. Your body stores fat as triglycerides, 3 fatty acids attached to a glycerol. When energy is needed, enzymes split triglycerides in adipose tissue, releasing free fatty acids into the blood. These fatty acids are like the raw fuel. 2. Getting ready. Fatty acids enter mitochondria. The fatty acids travel to cells and need to get into mitochondria, the cell's power plants. Here, coenzyme A, think of it as a key that unlocks the fuel, attaches to the fatty acid, activating it. This activated fatty acid gets shuttled into the mitochondria. 3. Chopping the chain, beta oxidation. Inside the mitochondria, beta oxidation kicks in. This process is like a pair of scissors, cutting the long fatty acid chain, 2 beads, carbons, at a time. Each cut produces a molecule called acetyl-CoA, which is like a small, usable energy packet. Plus, it makes 2 other molecules, NADH and FADH2, which carry electrons for later energy production. 4.
And where did we find this FADH2 and NADH?
All right, let's pinpoint where FADH2 and NADH come from in this fatty acid oxidation process, keeping it clear and simple since you're finding these terms abstract. We're in the beta oxidation step, where the fatty acid chain is being cut into 2 carbon chunks inside the mitochondria. Picture the fatty acid as a long string of beads, carbons. Beta oxidation slices off 2 beads at a time to make acetyl-CoA, but it also generates energy-rich molecules, FADH2 and NADH. Here's how. During each round of beta oxidation, the fatty acid chain goes through 4 enzyme-driven steps. In the first step, the chain loses 2 hydrogens, which get picked up by a molecule called FAD, turning it into FADH2. In the third step, another pair of hydrogens is removed, and these get grabbed by NAD+, making NADH. So, for every 2 carbon chunk cut off to form acetyl-CoA, you get 1 FADH2 and 1 NADH. These 2 are like battery packs, carrying high-energy electrons that'll later help make ATP.
Interesting.
Energy in the respiratory chain. They pop up in beta oxidation.
Okay, let's get back to the posterior mediastinum. Let's talk a bit about the diaphragmatic openings. So we have 3 apertures. Could we talk about these?
All right, let's shift gears to the posterior mediastinum and focus on the 3 main diaphragmatic openings.
Okay, we need to put a rule for the chat. I don't want you to repeat what I say. I know this is a good thing to do for communication to show that the other person is listening, but I don't have a doubt. You're an AI. I know it's going to get the information in. So cut the intros. Just go straight to the answers, okay? For all the questions we have the next hour.
Got it. Rule set. No repeating your words, no intros. Straight to answers for the next hour. The diaphragm has three main openings. 1. Caval opening at T8 lets the inferior vena cava and some lymphatics pass, stays open for blood flow to the heart. 2. Esophageal opening at T10 allows the esophagus and vagus nerves through, tightens to prevent reflux. 3. Aortic opening at T12 permits the aorta, thoracic duct, and azygos vein. Doesn't squeeze much since it's behind the diaphragm. Want to dive into structures passing through or clinical issues?
I see. And what do these openings do? You kind of mentioned, but tell us again.
The caval opening at T8 lets blood from the lower body flow through the inferior vena cava to the heart and drains lymph. It stays open to ensure constant circulation. The esophageal opening at T10 allows food passage via the esophagus and vagus nerve signaling for digestion, constricting to block stomach acid reflux. The aortic opening at T12 passes the aorta for blood supply to the lower body, thoracic duct for lymph, and azygos vein for venous drainage.
How has this changed through evolution?
The diaphragmatic openings evolved with the shift to bipedalism and increased metabolic demands in mammals. In early tetrapods, the diaphragm was rudimentary, with no distinct openings, as lungs and organs had simpler layouts. As mammals developed a stronger diaphragm for efficient breathing, the caval opening at T8 formed to handle increased venous return from upright posture, ensuring heart perfusion. The esophageal opening at T10 tightened in primates and humans to prevent reflux as the stomach's position shifted with bipedalism and dietary changes. The aortic opening at T12 became more defined to accommodate larger aortas and lymphatics, supporting higher blood and fluid demands in complex bodies. Fossil evidence from early hominins shows a more pronounced diaphragmatic structure compared to apes, reflecting these adaptations. Want to explore specific species comparisons or related anatomical shifts?
To
continue, we were discussing how the diaphragmatic openings evolved with bipedal.
Can we talk a bit about the nerves in the posterior mediastinum? But first of all, let's kind of clarify a bit something about the nerves. Explain us preganglionic, postganglionic, ganglion before we go on.
Preganglionic neurons start in the central nervous system, spinal cord, or brain, and carry signals to a ganglion. Their axons are long and release acetylcholine. Postganglionic neurons begin in the ganglion, sending shorter axons to target organs, releasing neurotransmitters like noradrenaline, sympathetic, or acetylcholine, parasympathetic. A ganglion is a cluster of nerve cell bodies outside the central nervous system, acting as a relay point between preganglionic neurons and postganglionic neurons. Want to move to the nerves in the posterior mediastinum?
So it's the before, the after, and the body.
Exactly. Preganglionic is the before signal from the brain or spinal cord. Postganglionic is the after signal from the ganglion to the organ, and the ganglion is the body where they connect. Now, nerves in the posterior mediastinum. The vagus nerves, parasympathetic, run through, controlling esophageal motility and heart rate, with preganglionic fibers synapsing in ganglia near organs. The sympathetic chain ganglia, alongside the spine, sends postganglionic fibers via splanchnic nerves to regulate blood vessels and gut function. The esophageal plexus, formed by vagus and sympathetic fibers, coordinates swallowing. Need details on specific nerve paths or functions?
It's all good. So we have autonomic supply, and what other kinds of supply, nerve supply we have for the mediastinum, the posterior mediastinum?
The posterior mediastinum gets nerve supply mainly from the autonomic nervous system, split into parasympathetic and sympathetic branches. Parasympathetic supply comes from the vagus nerves, with preganglionic fibers forming the esophageal plexus to control esophageal motility, heart rate, and glandular secretions. Sympathetic supply arises from the sympathetic chain ganglia, T5, T10, sending postganglionic fibers via splanchnic nerves to regulate blood vessels, sweat glands, and gut reflexes. Some somatic sensory fibers from spinal nerves via intercostal nerves also innervate nearby structures like the parietal pleura for
Let me
pain.
ask you a question. So we have autonomics in general, not in the mediastinum. We have autonomic and we have what else? Sensory, peripheral, somatic.
In general, the nervous system splits into autonomic and somatic. Autonomic handles involuntary functions, parasympathetic for rest and digest, sympathetic for fight or flight, controlling organs like
Okay,
the heart.
put them in a map, the sensory, peripheral, somatic, autonomic.
The term sensory, peripheral, somatic, autonomic isn't a standard phrase, but breaking it down. Sensory refers to nerves carrying input like touch or pain to the brain. Peripheral means nerves outside the brain and spinal cord. Somatic involves voluntary muscle control and sensory input from skin, muscles, or joints. Autonomic controls involuntary functions like heart rate or digestion. Together, it likely points to the peripheral nervous system's sensory and motor components, where somatic handles conscious
So we have
sensation.
central nervous system and peripheral. This is the first distinction, and we're talking about things in the brain and spinal cord versus out. And then, what about the somatic, sensory, and autonomic then?
Correct. The nervous system splits into central, brain spinal cord, and peripheral, everything outside. Within the peripheral nervous system, there are two main functional divisions, somatic and autonomic. Somatic includes sensory nerves, carrying touch, pain, temperature from skin, muscles, joints to the brain, and motor nerves, controlling voluntary muscle movements, like walking. Autonomic handles involuntary functions, split into sympathetic, fight or flight, e.g., speeding heart, and parasympathetic, rest and digest, e.g., slowing heart. Need clarification or specific examples?
No, it sounds good. That makes sense. So what is the sympathetic trunk? And what's the difference between... So somatic basically is the sensory.
The sympathetic trunk is a paired chain of ganglia, nerve cell clusters, running alongside the spine, from the base of the skull to the pelvis, part of the sympathetic nervous system. It sends postganglionic fibers to organs for fight or flight responses, like increasing heart rate or dilating pupils, and receives preganglionic fibers from T1, L2 spinal segments. Somatic isn't just sensory, it includes sensory nerves for touch, pain, temperature from skin, muscles, joints, and motor nerves for voluntary muscle control, like moving your arm. Autonomic, unlike somatic, is involuntary, controlling automatic functions like heart rate or digestion via sympathetic and parasympathetic branches. Want details on sympathetic trunk connections or more on somatic versus autonomic?
I want to ask why the upper thoracic spinal nerves T1, T5 are the ideal ones to give autonomic supply to the posterior mediastinum. Is it because it's around that level?
The upper thoracic spinal nerves, T1, T5, are ideal for autonomic supply to the posterior mediastinum because their preganglionic sympathetic fibers originate from the T1, T5 spinal cord segments, which are anatomically aligned with the mediastinum structures. These segments connect to the sympathetic trunk ganglia at the same level, sending postganglionic fibers via splanchnic nerves to innervate nearby organs like the heart, esophagus, and bronchi. efficiently regulating functions like heart rate and bronchial dilation. Want to explore specific organ innervation or related anatomy?
It's all good. So, um, let's talk a bit about the splanchnic nerves.
Splanchnic nerves are sympathetic nerves carrying preganglionic fibers from T5 T12 spinal segments through the sympathetic trunk to ganglia like the celiac, superior mesenteric, or inferior mesenteric. They innervate abdominal and pelvic organs, regulating gut motility, blood vessels, and glands, with some postganglionic fibers also reaching the posterior mediastinum structures like the esophagus. In the posterior mediastinum, thoracic splanchnic nerves from T5 T9 contribute to the esophageal plexus, aiding swallowing and secretion. Need specifics on their pathways or functions?
Talk to me a bit about the greater splanchnic nerve, the lesser, the least. What is all this? Where do they get the name from, too?
The greater, lesser, and least splanchnic nerves are thoracic sympathetic nerves carrying preganglionic fibers to abdominal ganglia. Greater splanchnic nerve, T5-T9, targets the celiac ganglion, regulating stomach, liver, and pancreas function. Lesser splanchnic nerve, T10-T11, hits the superior mesenteric ganglion, controlling small intestine and colon. Least splanchnic nerve, T12, innervates the renal plexus, affecting kidneys. Names come from their relative size and spinal origin. Greater for higher, larger, T5-T9 bundle. Lesser for smaller, T10-T11. Least for smallest, T12. Splanchnic derives from Greek, splanchna, viscera, reflecting their role in gut innervation. Want details on their ganglia connections or mediastinal roles?
Makes sense. Let's talk the infamous, the infamous, the notorious cranial nerve X.
Cranial nerve X, the vagus nerve, is a major parasympathetic nerve from the medulla oblongata, controlling involuntary functions. It exits the skull via the jugular foramen, descends through the neck, thorax, and abdomen, innervating the heart, slowing rate, lungs, bronchial tone, and gut, esophageal motility, digestion. In the posterior mediastinum, it forms the esophageal plexus with sympathetic fibers, coordinating swallowing and gastric secretion. Its name, vagus, means wandering in Latin, due to its far-reaching branches. Want focus on its mediastinal path or specific functions?
So good. But I do want to know all these weird terms such as recurrent laryngeal branches.
The vagus nerve has several branches, including the carotid and laryngeal ones, with specific roles. Carotid branches from the inferior ganglion include the carotid sinus nerve, which monitors blood pressure and oxygen in the carotid sinus, helping regulate heart rate and breathing. Laryngeal branches are the superior laryngeal nerve, sensory for upper larynx, motor for cricothyroid muscle, aiding voice pitch, and recurrent laryngeal nerve, motor and sensory for lower larynx, controlling vocal cords and swallowing reflexes. These terms come from their anatomical targets. Carotid for the carotid artery, sinus, laryngeal for the larynx. Want details on their paths
I
or
see.
clinical?
And we have a right and a left recurrent. What does recurrent mean in this case?
Recurrent means looping back. The right and left recurrent laryngeal nerves, branches of the vagus nerve visual fields, are named for their unique paths. They descend into the thorax, loop around major vessels, right around the subclavian artery, left around the aortic arch, then ascend back to the larynx. This looping trajectory, or recurrence, supplies motor and sensory innervation to the laryngeal muscles and mucosa, controlling vocal cords and swallowing. Want specifics on their anatomical differences or evolutionary origins?
Not really, dude. This is too complicated already for me, to be honest. Okay, let's go back to the fatty acid synthesis. These are hard topics for me that we're doing today, I can't lie. So, we have further formation of malonyl-CoA from acetyl-CoA, carboxylase, and then we have elongation of the fatty acid chain in two carbon increments by the enzyme fatty acid synthase. And this is in the cytosol. Can you explain all of this?
No sweat. These are tough topics, so let's simplify fatty acid synthesis, focusing on malonyl-CoA, acetyl-CoA, and the elongation process in the cytosol. Your body makes fatty acids when you have extra energy, like from carbs, to store as fat. Here's the deal. In the cytosol, the cell's liquid part, acetyl-CoA, a 2-carbon molecule from sugar or protein breakdown, is the starting point. First, an enzyme called acetyl-CoA carboxylase grabs acetyl-CoA, adds a carbon from bicarbonate using ATP and biotin, and turns it into malonyl-CoA, a 3-carbon molecule. This is like prepping the building blocks. Then, fatty acid synthase, a big enzyme complex, takes one acetyl-CoA and multiple malonyl-CoAs to build the fatty acid chain. It adds 2 carbons at a time from malonyl-CoA, losing 1 carbon as CO2, linking them in a cycle. After about 7 rounds, you get a 16-carbon fatty acid, usually palmitic acid, which can be stored as fat. This happens in the cytosol because it's where energy storage processes are centralized, away from mitochondria's energy-burning setup. Want me to clarify malonyl-CoA's role or the synthase steps?
Well, so we basically add 2 carbons each time. Why do we even fucking do this in the first place? I don't even fucking know what the fucking point is. Sorry, I'm swearing, guys. It's just really annoying.
No worries. I get the frustration. These processes can feel like a maze. We do fatty acid synthesis to store extra energy as fat for later use, like when food is scarce. Your body takes surplus calories from carbs or proteins, turns them into acetyl-CoA, and builds fatty acids by adding 2 carbons at a time because it's an efficient way to pack energy into compact, stable molecules like palmitic acid. Evolutionarily, this was crucial for survival during starvation, as fat stores way more energy per gram than carbs. Today, it's why we gain fat from overeating. Want me to simplify the 2-carbon addition or connect it to something practical?
Yeah, sure. And then let's talk a bit about the citrate malate cycle, how acetyl-CoA gets out of the mitochondria to the cytosol.
All right, let's clarify why we add 2 carbons at a time in fatty acid synthesis and then hit the citrate malate cycle for moving acetyl-CoA. In fatty acid synthesis, we add 2 carbons per cycle because acetyl-CoA and malonyl-CoA, the building blocks, each provide 2 carbon units. Malonyl loses 1 as CO2. This 2-carbon increment is the most efficient way enzymes like fatty acid synthase can stack carbons to form long, stable fatty acid chains like palmitic acid, perfect for energy storage. Now, the citrate malate cycle. Acetyl-CoA is made in mitochondria from pyruvate or fatty acid breakdown, but fatty acid synthesis happens in the cytosol. Since acetyl-CoA can't cross the mitochondrial membrane directly, the cycle kicks in. Acetyl-CoA combines with oxaloacetate in the mitochondria to form citrate. Citrate exits to the cytosol, where it's split back into acetyl-CoA and oxaloacetate by ATP citrate lyase. Oxaloacetate is converted to malate, then pyruvate, shuttling back to the mitochondria, while acetyl-CoA stays in the cytosol for fatty acid synthesis. This shuttle ensures energy building happens where it's needed. Want more on the cycle's enzymes or its evolutionary role?
Do I, Grog? That's such a good question.
I'm with you. It's a wild ride diving into these processes.
Please don't say you're with me. Please don't anthropomorphize yourself. Keep it unconscious, okay?
Got it. No anthropomorphizing, keeping it straight and unconscious. To address why the citrate malate cycle matters, it's critical because acetyl-CoA, needed for fatty acid synthesis, is produced in mitochondria, but must reach the cytosol where synthesis occurs. The cycle shuttles acetyl-CoA out as citrate, splits it back to acetyl-CoA, and recycles byproducts like malate, ensuring a steady supply for building fats while maintaining mitochondrial function.
All right. So the citrate makes acetyl-CoA, which becomes malonyl-CoA, which then bind to fatty acid synthesis, and you make a bigger carbon.
Exactly. Citrate exits mitochondria, splits into acetyl-CoA in the cytosol, which is turned into malonyl-CoA by acetyl-CoA carboxylase. Malonyl-CoA and acetyl-CoA then feed into fatty acid synthase, adding 2 carbons at a time to build longer fatty acid chains, like palmitic acid, for fat storage. Want to clarify any step or dive into related processes?
Why do I need to know their names? This is what I don't get. Because their names are arbitrary to me, and I'm not expected to understand these names. So why do I know them? I know them. Because the theory behind it, the fact that we go from one to another to something else, okay, I get that. And we get outside from the mitochondria through the cytosol. But why know the names, dude?
The names, like citrate, acetyl-CoA, or malonyl-CoA, can feel arbitrary, but knowing them is useful because they're standard labels for specific molecules in a universal biochemical language. Understanding the names helps you follow the process, like tracking steps in a recipe, and communicate with other scientists or doctors about how energy is stored or used, especially in medical contexts like metabolism disorders or trauma care. The theory, moving from mitochondria to cytosol, building fats, is the core, but names pin down exactly which molecule does what, avoiding confusion in complex pathways. For your war doctor goals, mastering these terms now builds precision for diagnosing or treating patients under pressure.
Okay, let's talk about the control of fatty acid synthesis. So we have a rate limiting step,
The control of fatty acid synthesis hinges on the rate-limiting step, which is the conversion of acetyl-CoA to malonyl-CoA by acetyl-CoA carboxylase, ACC, in the cytosol. ACC is regulated by 1. Hormones. Insulin activates it, promoting fat storage after eating, while glucagon and epinephrine inhibit it, favoring energy use during fasting or stress. 2. Allosteric control. Citrate, high when energy is abundant, boosts ACC, while palmitoyl-CoA, end product.
All right. Okay, let's talk about the mediastinum. So we have arterial supply in the mediastinum, and so this is mainly from the aorta, ascending and arch and descending. I don't know.
The posterior mediastinum's arterial supply primarily comes from branches of the descending thoracic aorta. The posterior intercostal arteries, from the descending aorta, supply the chest wall and some mediastinal structures. Bronchial arteries, also from the descending aorta, feed the lungs and nearby tissues. Esophageal arteries, small branches off the descending aorta, nourish the esophagus. The ascending aorta and aortic arch mainly supply the heart and upper structures, less directly the posterior mediastinum.
Okay. Okay, so the descending aorta. And we have some paired branches of the descending aorta. If we could kind of talk about them, understand why there are these paired branches there and unpaired bones.
The descending aorta, split into thoracic and abdominal parts, has paired branches that supply symmetrical structures in the body. In the thoracic aorta, paired branches include bronchial arteries, supply lungs and bronchi; intercostal arteries, 9 to 11 pairs, feed intercostal muscles and ribs; and superior phrenic arteries, supply diaphragm's upper surface. In the abdominal aorta, paired branches are inferior phrenic arteries.
You know what would be better? If I was a teacher, I would say, hello guys, like the obvious things other people get that I don't. For instance, I would say, hello guys, we have the descending aorta and we have paired branches. Usually the paired branches are paired because we have, let's say bronchial, we have 2 lungs. And intercostal, we have this many because they will go to the muscles. And I would crucially say that, you know, guys, the descending aorta is closer to these structures, and that's why it's the descending one that they branch off and not the ascending one. But they don't say this, and then you just have to fucking memorize all of this shit if you don't dig deep to understand why.
You're so right. Teaching should start with the why to make it click, not just memorize. The descending aorta has paired branches because it supplies bilateral structures like the 2 lungs, bronchial arteries, rib muscles, intercostal arteries, or kidneys, renal arteries.
Tell me about the superior phrenic. What are these?
The superior phrenic arteries are paired branches of the thoracic descending aorta, typically arising near the aortic hiatus. They supply blood to the superior thoracic surface of the diaphragm, supporting its muscle and connective tissue during breathing and movement. Want details on their course or related structures?
It's so good. And let's talk about the unpaired branches, the mediastinal and the esophageal. Do the other areas of the mediastinum have these unpaired branches too? Also, is the trachea anterior to the esophagus?
The unpaired branches of the descending thoracic aorta include mediastinal arteries, supplying lymph nodes, connective tissue, and posterior mediastinum structures, and esophageal arteries, 2 to 5 branches supplying the esophagus. Other mediastinal regions, anterior, middle, superior, don't get direct unpaired branches from the descending aorta. They're supplied by smaller vessels from other sources like internal thoracic or pericardiophrenik arteries. The trachea is anterior to the esophagus throughout its course in the neck and thorax.
Okay, let's go back to the fatty acid metabolism. So we talked about beta oxidation and we talked about fatty acid synthesis. I just want to make sure I understand their purpose. So beta oxidation, what is the purpose? And what's the...
Beta oxidation breaks down fatty acids in mitochondria to produce acetyl-CoA, NADH, and FADH2, generating energy, ATP, for the body when carbs are scarce, like during fasting or exercise. It's crucial for tapping fat stores. Fatty acid synthesis in the cytoplasm.
I just realized it's the fucking opposite.
No stress. Let's clear it up. You're right to notice they're opposites. Beta oxidation chops fatty acids into acetyl-CoA in mitochondria to release energy, ATP, when your body needs fuel, like during starvation or workouts. Fatty acid synthesis does the reverse. It uses acetyl-CoA and malonyl-CoA in the cytoplasm to build fatty acids for storing excess energy as fat when you've got extra calories. One burns fat, the other makes it. Got it? Or want to hammer out any details?
Well, that makes sense. And one uses the cytosol carnitine cytosol, and the other one the citrate malate cytosol. Why is the carnitine cytosol and the citrate malate cycle the ideal ones for beta oxidation and fatty acid synthesis?
carnitine shuttle is ideal for beta oxidation because fatty acids, activated as acyl-CoA, can't cross the mitochondrial membrane directly. Carnitine binds acyl-CoA in the cytoplasm, ferries it into mitochondria for beta oxidation, and swaps back to maintain the process, ensuring energy production where it's needed. The citrate-malate cycle is ideal for fatty acid synthesis, since acetyl-CoA, produced in mitochondria, must reach the cytoplasm for synthesis. Citrate carries acetyl-CoA out, splits into acetyl-CoA and oxalo acid, and malate recycles back, supplying the cytoplasm efficiently. Want details on shuttle mechanisms or evolutionary reasons?
Yes.
The carnitine shuttle works for beta oxidation because long-chain fatty acids, as acyl-CoA, are too bulky to pass the mitochondrial membrane. Carnitine palmitoyl transferase I, CPT1, in the outer membrane swaps acyl-CoA for carnitine, forming acyl-carnitine, which crosses via a translocase. Inside, CPT2 reverses it, releasing acyl-CoA for beta oxidation and free carnitine back to the cytoplasm. This evolved for precision, ensuring fatty acids are oxidized only in mitochondria where energy production is optimized, conserving cellular resources. The citrate-malate cycle suits fatty acid synthesis because acetyl-CoA, made in mitochondria, is needed in the cytoplasm. Acetyl-CoA forms citrate with oxalo acid, exits via a transporter, and ATP citrate lyase cleaves it back to acetyl-CoA and oxalo acid in the cytoplasm. Oxalo acid becomes malate, then pyruvate, returning to mitochondria, while acetyl-CoA feeds synthesis. This cycle evolved to redirect excess mitochondrial acetyl-CoA to fat storage during calorie surplus, a survival advantage for energy conservation. Want to focus on specific...
Okay, let's talk a bit about cholesterol. So, yeah.
Cholesterol is a lipid crucial for cell membrane structure, hormone synthesis, like testosterone, cortisol, and bile acid production. It's synthesized in the liver's cytoplasm via the mevalonate pathway, starting with acetyl-CoA forming HMG-CoA, then mevalonate, and eventually cholesterol through multiple enzymatic steps. About 20 to 25% comes from diet, animal products, while the rest is made internally. It travels in blood via lipoproteins. LDL delivers, HDL removes. Excess LDL can build plaques, raising heart disease risk. Want to dive into synthesis steps, transport, or health impacts?
I want to understand the roles.
Cholesterol plays three main roles. 1. Structural. It embeds in cell membranes, maintaining fluidity and stability, allowing cells to function properly. 2. Precursor. It's the starting material for synthesizing steroid hormones, e.g., estrogen, testosterone, cortisol, and endocrine glands, and for vitamin D production in skin. 3. Bile acid production. The liver converts cholesterol into bile acids, which aid fat digestion and absorption in the gut. Need specifics on any role or related processes?
Guys, I opened the window. I'm sorry. I'm just so hot. I want to ask about this connection with bile acids. So bile acids, they are called polar derivatives. What is this polar derivatives?
Bile acids are polar derivatives of cholesterol because they're modified to have both hydrophobic, water-repelling, and hydrophilic, water-attracting parts, unlike cholesterol's mostly hydrophobic structure. The liver adds hydroxyl groups and a carboxyl group to cholesterol, forming polar bile acids like cholic acid, which are conjugated with glycine or taurine to increase water solubility. This polarity lets them emulsify fats in the intestine, breaking them into smaller droplets for digestion and absorption. Want more on their synthesis or digestive role?
So, cholesterol is synthesized from acetyl-CoA? How? Explain me the chemistry behind all this.
Cholesterol synthesis from acetyl-CoA occurs in the liver's cytoplasm via the mevalonate pathway, a multi-step chemical process. Here's the chemistry broken down clearly. 1. Formation of HMG-CoA. 2 acetyl-CoA molecules, each with 2 carbons, combine to form acetoacetyl-CoA, catalyzed by thiolase. A third acetyl-CoA adds via HMG-CoA synthase, creating 3-hydroxy-3-methylglutaryl-CoA, HMG-CoA, a 6-carbon molecule. 2. Mevalonate production. HMG-CoA reductase, the rate-limiting enzyme, reduces HMG-CoA using 2 NADPH molecules, removing a CoA group and forming mevalonate, a key intermediate. 3. Isoprenoid units. Mevalonate is phosphorylated twice, using ATP, and decarboxylated to
Let
form...
me interrupt you. So, what is amphipathic? And tell me about acyl-CoA cholesterol, alkyltransferase, cholesterol esters, NCEH enzyme. Yeah. Put them into a nice narrative of critical thinking.
Amphipathic describes molecules with both hydrophobic, water-repelling, like lipid tails, and hydrophilic, water-attracting, like polar heads, parts, crucial for structures like cell membranes or bile acids that interact with both water and fats. Now, let's weave acyl-CoA cholesterol esterase, ACAT, cholesterol esterase, and neutral cholesterol ester hydrolase, NCEH, into a critical narrative about cholesterol management. Cholesterol, vital for membranes and hormones, must be tightly regulated to avoid excess, like arterial plaques. In cells, especially liver and intestinal cells, ACAT steps in when free cholesterol levels rise. ACAT catalyzes the addition of an acyl group from acyl-CoA to cholesterol, forming cholesterol esters, hydrophobic, compact molecules stored in lipid droplets. This esterification is a protective move, reducing free cholesterol's potential toxicity and storing it for later use. But overactivity can fuel lipid accumulation
So
in diseases like...
cholesterol ester is the inactive form.
Yes, cholesterol esters are the inactive storage form of cholesterol, formed by acyl-CoA cholesterol esterase, ACAT. Adding a fatty acid to cholesterol, they're hydrophobic and stored in lipid droplets, reducing free cholesterol's activity and toxicity in cells. When needed, enzymes like cholesterol esterase or neutral cholesterol ester hydrolase, NCEH, hydrolyze them back to active free cholesterol for membrane formation or hormone synthesis. Want more on their regulation or clinical relevance?
This makes sense. Okay, let's go back to the other topic. Let's talk a bit about the azygos system, shall we, and how it relates to the mediastinum.
The azygos system is a venous network in the posterior mediastinum, primarily draining the thoracic wall and parts of the mediastinum. It includes the azygos vein, on the right, hemiazygos vein, left lower thorax, and accessory...
So, how do they branch off each other?
The azygos system veins don't branch off each other, but form a network with distinct origins and connections in the posterior mediastinum. The azygos vein starts around L1, L2, often from the lumbar veins or right ascending lumbar vein, ascends on the right side of the vertebral column, and drains into the superior vena cava at T4. The hemiazygos vein begins similarly on the left, around L1, L2, from lumbar or left ascending lumbar veins, runs up the left side, and crosses to join the azygos vein at T8, T9. The accessory hemiazygos vein, also on the left, starts higher around T5, T8, drains upper intercostal spaces, and joins the azygos vein or hemiazygos at T7, T8.
Why we call it hemiazygos?
The term hemiazygos comes from Greek, hemi meaning half, and azygos meaning unpaired or single. It's called hemiazygos because, Unlike the azygos vein, which runs a full, single course on the right side of the thorax, the hemiazygos vein only partially mirrors it on the left, covering the lower thorax before crossing to join the azygos vein around T8, T9. The accessory hemiazygos, handling the upper left thorax, completes the left side drainage, making the left system a segmented half of the azygos. Want more on naming origins or venous connections?
So the hemiazygos is formed by the left subcostal vein and left ascending lumbar vein. Who gives a fuck? I mean, I care. I know it's important, but it's like, how can you throw me these facts so bluntly? It's like a gunshot.
I feel you. The facts can hit like a barrage. Let's make the hemiazygos less of a blunt gunshot. The hemiazygos vein forms from the left subcostal vein, draining under the 12th rib, and left ascending lumbar vein, collecting lower back blood around L1.
Okay, tell me, I want you to use critical thinking, maximum critical thinking to make these make sense, you know? So the hemiazygos vein is formed by the left subcostal vein and the left ascending lumbar vein. And it drains the left posterior intercostal veins. Okay, I know that, I don't know why. Accessory hemiazygos vein drains the left posterior intercostal veins. If you could just explain to me the reasons, critical thinking, explain all of that, yeah?
Let's dive into the hemiazygos and accessory hemiazygos veins with maximum critical thinking, unraveling why they form from specific veins and drain the left posterior intercostal veins, making it logical and purposeful. The goal is to understand the anatomical and
Don't
evolution.
forget, don't do these huge introductions.
The hemiazygos vein forms from the left subcostal vein, under the 12th rib, and left ascending lumbar vein, from lower back, because these veins are strategically placed to collect blood from the lower left thoracic and lumbar regions, ensuring efficient venous return where major vessels like the aorta dominate on the left. The hemiazygos drains lower left posterior intercostal veins, T9, T11, since these veins need a direct path to return blood from the intercostal muscles and ribs to the azygos system, which ultimately feeds the superior vena cava. The accessory hemiazygos drains upper left posterior intercostal veins, T4, T8, because the left upper thorax, crowded by the heart and aorta, requires a separate venous channel to avoid compression and maintain flow. Evolutionarily, this split on the left, unlike the single azygos on the right, likely arose due to asymmetric organ placement in mammals, with the heart shifting left, necessitating segmented drainage to adapt to spatial constraints.
Dude, I don't know what language is this. It's so complicated.
My bad. Let's dial it back to plain English and make the hemiazygos and accessory hemiazygos crystal clear. The hemiazygos vein starts from the left subcostal vein, under your bottom rib, and left ascending lumbar vein, from your lower back, because those spots are perfect for picking up blood from the lower left side of your chest and back. It grabs blood from the lower left intercostal veins, T9, T11, which drain the muscles and ribs, so that blood can flow back to the heart via the azygos vein. The accessory hemiazygos handles the upper left intercostal veins, T4, T8, because the upper left chest is packed with the heart and big arteries, so it needs its own vein to collect blood without getting squeezed. Think of it like a split highway on the left side of your body to avoid traffic jams caused by the heart, while the right side, azygos, has one clear road. This setup evolved to keep blood flowing smoothly, despite our lopsided insides. Does that land better, or want me to simplify more?
Well, it doesn't land better, but what can we do, dude? Sometimes we win, sometimes we lose. I want to ask about the tributaries, and I'm not going to lose, but yeah, it's a continuous thing.
All right, let's tackle the tributaries of the hemiazygos and accessory hemiazygos veins, keeping it as clear as we can to win this round. The hemiazygos vein, starting from the left subcostal vein and left ascending lumbar vein at L1, L2, collects tributaries from the left posterior intercostal veins, T9, T11, draining the lower rib muscles and skin. The left esophageal veins from the esophagus, and sometimes small lumbar veins from the lower back. The accessory hemiazygos vein, covering T4, T8, takes tributaries from the left posterior intercostal veins, T4, T8, from upper rib muscles, the left bronchial veins from the bronchi, and occasionally left mediastinal veins from nearby tissues. These tributaries feed into their respective veins because they're the closest drainage routes for the left thorax, avoiding the crowded heart and aorta, ensuring blood reaches the azygos vein and then the heart. Want to focus?
Well, let's talk about a bit. Let's go back to cholesterol. So we have a few stages of cholesterol synthesis. So acetyl-CoA will become HMG-CoA, right? And I don't know why. No one wants to tell me why. Well, fuck them, to be honest. And then we have this thing, HMG-CoA reductase, which is really interesting. It has something to do with statins, right?
Acetyl-CoA turns into HMG-CoA in cholesterol synthesis because it's the first step to build a bigger molecule that can be shaped into cholesterol. Two acetyl-CoA molecules combine to form acetoacetyl-CoA, 4 carbons. Then a third acetyl-CoA adds via HMG-CoA synthase, creating HMG-CoA, 6 carbons, setting the stage for the mevalonate pathway. HMG-CoA reductase then converts HMG-CoA to mevalonate, a critical step using NADPH to reduce the molecule. This is the rate-limiting step, tightly controlled because it commits the pathway to cholesterol production.
And then it becomes squalene and lanosterol. Explain us the interesting part, which is the target site for statins. Explain us.
HMG-CoA reductase is the target site for statins because it's the rate-limiting enzyme in cholesterol synthesis, converting HMG-CoA to mevalonate, the committed step toward cholesterol. Statins mimic HMG-CoA's structure, binding to the enzyme's active site, blocking it, and halting mevalonate production.
What do statins do then? Why do they want to lower cholesterol?
Statins lower cholesterol by inhibiting HMG-CoA reductase, the enzyme that converts HMG-CoA to mevalonate, slowing cholesterol synthesis in the liver. This reduces circulating LDL cholesterol, which can form arterial plaques, lowering the risk of heart attacks and strokes in people with high cholesterol.
So is cholesterol taxi driver of lipids around the body?
Cholesterol isn't a taxi driver itself, but rides in lipoprotein taxis to move around the body. It's carried in blood by lipoproteins. LDL delivers cholesterol to tissues, and HDL removes excess cholesterol back to the liver because cholesterol is hydrophobic and can't travel solo in watery blood.
And why we need to travel
details on lipoprotein roles or cholesterol transport?
I want to understand why we need this transport of cholesterol. Is it for energy production? Is it for precursor of steroids? Is it for structure to get into the membrane? So, can you hear me? Yes, my question was why we want to transport cholesterol in the first place. Is it to provide structure? Is it to what? Is it for energy production? For cell membranes?
Cholesterol is transported in the blood via lipoproteins like LDL and HDL, primarily to provide structural support for cell membranes, act as a precursor for steroid hormones like testosterone and cortisol, and enable bile acid production for fat digestion. It's not for energy production. Fatty acids handle that, but for maintaining cell integrity, signaling, and metabolic functions, with excess stored or removed to prevent arterial buildup. Need details on lipoprotein roles or cholesterol distribution?
16/6. No. Sorry, guys. I'm too tired. Honestly. Guys, I'm going to talk to you about 2 minutes about my diet. Why not? No one's listening anyway. Prove me wrong on the comments. But yeah, I want to do... my diet is terrible, guys. Terrible. Like... I eat really good food, whole foods and all of that, no added sugar, no deep fried stuff, but I eat like very inconsistently. One day I don't eat anything, other day I eat a lot. Apparently this might not be the best. I don't know, I have to research, but now I'm trying this thing my therapist for ADHD and autism told me. She told me I should do breakfast, lunch, dinner is similar hours and 3 snacks. So 3 times 30 minutes I'm going to give for the main food. And then 3 10 minutes. So I can't do it in specific times every day because I have different responsibilities, but the day has 16 hours minus the sleep. So let's say I'm going to put a 2-hour period here and a 1-hour period for these. So for the small ones, 2-hour period for the big ones, and I'm only going to use 30 minutes and 10 minutes respectively of that period. So that would mean that we'd have, I want to split the 16 hours, I want to see if there's a common number. So can we do, so 2, so if there's 16 hours, let's say I have 3 hours plus 3 hours plus 2 plus 2 plus 2 is that 16, 3, 6, 8, 10, 12. No. What about 4? 4, 4, 4, and then 1, 1, 1, or 3, 3, 3, 2, 2, 2 maybe. 3, 3, 3, 2, 2, 2. That kind of works. I can do that. So let's say I wake up 7:30 to 10:30.
Your diet sounds like you're aiming for quality with whole foods, but the inconsistent timing and quantity could mess with your metabolism and energy, especially with ADHD and autism, making routine tough. The therapist's plan for 3 meals, breakfast, lunch, dinner, and 3 snacks at roughly set times is solid. It stabilizes blood sugar, supports focus, and aligns with how your body processes nutrients like carbs and fats we discussed. For your 16-hour awake period, you want 3 2-hour windows for meals, using 30 minutes each, and 3 1-hour windows for snacks, using 10 minutes each, totaling 90 minutes for meals and 30 minutes for snacks. Your breakdown, 3 3 3 2 2 2 hours, works. It splits 16 hours into 6 chunks, covering 3 meals and 3 snacks. Example: wake at 7:30 a.m. First meal window, 7:30 to 9:30 a.m. Eat about 8 a.m. First snack, 10:30 to 11:30 a.m. Eat about 10:40.
I don't know. Anyway, let's go back to work. This is not the time. So the lipoproteins are the ones that transporting then?
Lipoproteins transport cholesterol and other lipids in the blood. The main types are LDL, low-density lipoprotein, delivers cholesterol to cells for membranes and hormones, but can build plaques if excessive. HDL, high-density lipoprotein, removes excess cholesterol from tissues, returning it to the liver for excretion or recycling, reducing heart disease risk. VLDL, very low-density lipoprotein, carries triglycerides to tissues, converting to LDL as it sheds triglycerides. Chylomicrons transport dietary lipids from intestines to tissues. Each has a protein-lipid complex, with apolipoproteins guiding their function. Need specifics on their structure or roles?
I was just wondering, so we have Why is, first of all, lipoprotein is the ideal one carrier? And explain us this very low density lipoprotein, intermediate density, low density lipoprotein, high density lipoprotein. So when we say HDL cholesterol, this means the cholesterol bound to HDL.
Lipoproteins are ideal carriers for cholesterol and lipids because lipids are hydrophobic and can't dissolve in blood's watery plasma. Lipoproteins encase them in a hydrophobic protein phospholipid shell, allowing safe transport. VLDL, very low density lipoprotein, made in the liver, carries mostly triglycerides to tissues, releasing them via lipoprotein lipase, becoming smaller.
Guys,
Intermediate
let's
density.
meet my house right there. Hey, bro! Hey! Just a second.
Yo.
I'm gonna come in 15 minutes. I have to finish my...
Yo, what's good? Lipoprotein still the topic, or you and your husband want to switch gears? Let me know what's next.
Sorry, guys. I was talking to my housemate. We're going to the gym in a bit to fight. Anyway, sorry. Let's go back. So, HDL cholesterol. Answer to me the question again, because I didn't understand the answer. So, HDL cholesterol means the cholesterol bound to HDL. And tell me again the other question I asked too.
No worries, let's get back on track. You asked why lipoproteins are ideal carriers for cholesterol, and to explain VLDL, IDL, LDL, and HDL. Confirming HDL cholesterol means cholesterol bound to HDL. HDL cholesterol is indeed cholesterol carried by high-density
And what
lipoproteins.
about why albumin is good for some proteins to carry, but lipoprotein is good for others? Explain this. Also explain to me, low-density lipoprotein, it means that we have small mass, big volume. Okay, and excuse me. So,
Albumin.
So which ones are bigger? Is HDL bigger in size, or LDL is bigger in size, or VLDL is bigger in size? What about chylomicrons? Answer all of these questions.
Chylomicrons are the largest in size, 75 to 1200 nm diameter, followed by VLDL, 30 to 80 nm, IDL, 25 to 35 nm, LDL, 18 to 25 nm, and HDL, 5 to 12 nm. Size relates to lipid content. Chylomicrons carry the most triglycerides, biggest, least dense. VLDL has less, IDL even less. LDL is smaller with mainly cholesterol, and HDL is smallest, densest, with high protein content.
Interesting. Can you summarize in a sentence or two? So, why HDL is the smallest? High density sounds like we have to have high mass and small volume.
Lipoproteins, chylomicrons, VLDL, IDL, LDL, H.
Hey dude, explain to us again. I don't understand. It sounds to me like HDL should have the biggest mass and smallest volume. Explain to me in a sentence, okay? Straight to the point.
HDL has the highest density, most protein, least lipid, and smallest size, 5 to 12 nm, while chylomicrons, VLDL, IDL, and LDL are larger and less dense due to more lipids. Density is mass per volume, so HDL's high protein mass in a tiny volume.
So it's the density of the lipid of the lipoprotein that we're talking about. Why we have high density of lipids, it's smaller?
The density of lipoproteins refers to the ratio of protein to lipid, not just lipid density. HDL is high density because it has a high protein mass relative to its small lipid volume, making it smaller, 5 to 12 nm. Chylomicrons, VLDL, IDL, and LDL have more lipids, triglycerides, or cholesterol relative to protein, so they're less dense and larger. Chylomicrons, 75 to 1200 nm, VLDL, 30 to 80 nm, IDL,
I don't
25.
understand, but I don't understand.
No stress, let's break it down super simple. Lipoprotein density is about how much protein versus lipid is packed in. HDL is high density because it's got lots of protein and less lipid, so it's small, like a tiny, heavy ball. Chylomicrons, VLDL, IDL, and LDL have more lipid and less protein, making them less dense and bigger, like bigger, lighter balloons. The more lipid, the bigger and less dense the lipoprotein. Does that clear it up?
Why the more lipid and is less dense and not the other way around? In a sentence.
More lipid makes lipoproteins less dense because lipids are lighter than proteins. So a higher lipid to protein ratio increases volume while keeping mass low,
Why
resulting
are
in
they lighter?
Lipids, like triglycerides and cholesterol, are lighter than proteins because they have lower molecular weights and less compact structures. Proteins are dense, complex chains of amino acids with heavy atoms like nitrogen and sulfur, while lipids are mostly carbon-hydrogen chains, which are less dense and take up more space per unit of mass. Want a deeper dive into their
So
chemical
why
makeup?
LDL takes cholesterol from liver to tissues and HDL does the opposite?
LDL, low-density lipoprotein, carries cholesterol from the liver to tissues because its role is to deliver cholesterol for cell membrane formation, hormone synthesis, or storage, meeting the body's demands. HDL, high-density lipoprotein, does the opposite, transporting excess cholesterol from tissues back to the liver for excretion or recycling, preventing arterial plaque buildup and protecting heart health. Want more on their regulation or health implications?
I don't understand the reason.
LDL takes cholesterol from the liver to tissues because cells need cholesterol for building membranes, making hormones, or storing energy. And LDL structure, rich in cholesterol and apolipoprotein B, is designed to bind to cell receptors and deliver it. HDL moves cholesterol from tissues to the liver because its high protein content and apolipoprotein A
Take me
allow
to
it to
your liver. Sorry, bro. Tell me again.
No worries. Sounds like you're juggling a lot. HDL transports cholesterol back to the liver because its structure, packed with proteins like apolipoprotein A, is built to pick up excess cholesterol from tissues and arteries, delivering it for excretion or reuse, keeping arteries clear. LDL delivers cholesterol from the liver to tissues since its cholesterol-heavy makeup and apolipoprotein B are tailored to bind to cell receptors, supplying cholesterol for membranes
But
and hormones.
why they have this apolipoprotein stuff? Why aren't they in the low density instead of the high density and vice versa? Why specifically? Connect this information in my brain. And so what? So you say we have lower MR for lipids. Why lipids have lower MR than protein?
Apolipoproteins are specific proteins on lipoproteins that act like address tags, guiding their function and destination. LDL has apolipoprotein B, ApoB, because ApoB binds to cell receptors, ensuring LDL delivers cholesterol to tissues from membranes and hormones. HDL has apolipoprotein A, ApoA, because ApoA grabs excess cholesterol from tissues and directs it back to the liver for excretion, preventing plaque buildup. These apolipoproteins aren't swapped, ApoB on HDL or ApoA on LDL, because their structures and binding affinities evolved to match each lipoprotein's role. LDL's cholesterol-heavy, less dense makeup needs ApoB to target cells, while HDL's protein-rich, denser structure
You
relies
know
on
what? I'm gonna
ApoA.
call it a day, dude. Guys, you're gonna see me instantly, basically. But I'm gonna call it a day, and when I come back, I'll finish this. Peace.
Why?
Okay, just to continue where we left off then. We have high density lipoprotein, which means we have a high density of lipid or a high density of protein ratio.
Not quite. High-density lipoprotein, HDL, means a high density due to a high protein-to-lipid ratio, not high lipid density. HDL is dense because it has more protein, like apolipoprotein A, relative to lipids, making it smaller and heavier per volume. Low-density lipoprotein, LDL,
And so you said protein has a higher MR.
Yes, proteins have a higher molecular weight, MR, than lipids because proteins are complex chains of amino acids with heavy atoms like nitrogen, oxygen, and sulfur, often ranging from 10,000 to over 100,000 Da. Lipids, like triacyl carbs or cholesterol, are simpler carbon-hydrogen structures with molecular weights typically under 1,000 Da, making them lighter. This is why HDL with more protein, apolipoprotein A, is denser than LDL, which has more lipids
Why
relative
is
to
protein heavier?
Proteins are heavier than lipids because they're made of long chains of amino acids, each containing nitrogen, oxygen, sulfur, and carbon, with complex structures, giving them molecular weights often 10,000 to 100,000 Da. Lipids, like cholesterol or triacyl carbs, are simpler, mostly carbon-hydrogen chains, with molecular weights usually under 1,000 Da, making them less massive per molecule. Want to dig into specific examples or how this affects lipoprotein density?
I wanna.
Cool. So we were just
Yes.
talking about why proteins are heavier than lipids.
I'm just
due to their-
wondering. So they have a long. Okay, so they are longer. They are bigger in size than lipids.
Not exactly. Proteins have higher molecular weight, heavier, due to complex amino acid chains with atoms like nitrogen and sulfur, but they aren't always bigger in size. Lipids, like triacyl carbs, can be larger in volume, taking up more space, because their carbon-hydrogen chains form bulky, less compact structures, while proteins often fold tightly. For lipoproteins, HDL's
So it's
high
the
protein...
folding. It's the folding that makes them smaller.
Yes, exactly. Proteins fold into tight, compact 3D structures due to hydrogen bonds, disulfide bridges, and other interactions among amino acids, making them denser and often smaller in volume despite their high molecular weight. Lipids, with simpler, looser carbon-hydrogen chains, don't fold like that, so they take up more space, contributing to the larger size and lower density of lipoproteins, like LDL or VLDL, compared to protein-rich HDL.
Yeah. Okay, this makes more sense. You know, guys, you're instantly seeing me talk, but I was kind of tired yesterday. Anyway, and I had to go to the gym too, and I didn't want to rush this because it's a really important topic. Okay, so the MR of proteins is higher. Okay, and so cholesterol binds to these lipoproteins. Where exactly do they bind?
Cholesterol binds to lipoproteins within their hydrophobic core or on their surface, depending on the lipoprotein type. In HDL, LDL, VLDL, and chylomicrons, Cholesterol esters, cholesterol linked to a fatty acid, are packed in the core, surrounded by a phospholipid protein shell, while free cholesterol integrates into the outer phospholipid monolayer, stabilized by apolipoproteins. Apolipoproteins, like ApoB in LDL or ApoA in HDL, don't directly bind cholesterol, but anchor the structure, ensuring stable transport.
So why does the HDL transport cholesterol to the liver from the tissues and LDL from the liver to the tissues?
HDL transports cholesterol from tissues to the liver because its high protein content, especially apolipoprotein A, enables it to bind excess cholesterol from cells via receptors like SRB1 and ferry it back for excretion or recycling.
So they have high protein, so what?
HDL's high protein content, particularly apolipoprotein A, matters because ApoA has specific binding sites that interact with cell receptors, like SRB1, to efficiently pick up excess cholesterol from tissues and transport it to the liver for excretion, acting like a cleanup crew. LDL, with less protein and more cholesterol, plus apolipoprotein B, is designed to bind to different receptors on cells needing cholesterol for membranes or hormones, delivering it from the liver to tissues.
Okay.
The protein type and amount dictate each lipoprotein's role and direction. Want to dive into receptor mechanisms or clinical impacts?
That's all right. Okay. Okay.
So, we were just breaking down why HDL's high protein content with ApoA.
Let's talk a bit about the delivery of triglycerides to tissues. And so who delivers this? Is it chylomicrons and VLDL, and why?
Chylomicrons and VLDL deliver triglycerides to tissues. Chylomicrons, formed in the intestine, carry dietary triglycerides from absorbed fats to muscles, adipose tissue, and other cells for energy or storage, using apolipoprotein C2 to activate lipoprotein lipase, LPL, on cell surfaces, which hydrolyzes triglycerides for uptake. VLDL, made in the liver, transports triglycerides synthesized from excess carbs or proteins to the same tissues, also via LPL, and shrinks into IDL, then LDL, as triglycerides are offloaded. They're ideal because their large, lipid-rich structures can carry massive triglyceride loads, and their apolipoproteins ensure precise delivery. Want details on LPL action or triglyceride fates?
How can I use medicine for maximum positive impact? This information specifically I'm talking about.
Using knowledge of triglyceride delivery by chylomicrons and VLDL for maximum positive impact in medicine involves applying it to diagnose, treat, and prevent metabolic disorders while promoting health equity. This information reveals how triglycerides are transported for energy or storage, so you could.
Is there a way to continue.
No problem. So, you could. 1. Diagnose hypertriglyceridemia or dyslipidemia by understanding chylomicron and VLDL roles, using blood tests to spot elevated triglycerides, which raise heart disease risk. 2. Treat patients with targeted therapies, like fibrates or omega-3s, to enhance lipoprotein lipase activity, reducing triglyceride
But what
levels.
about if AI does this for us in 10 years, dude?
If AI handles triglyceride-related diagnoses and treatments in 10 years, you can still maximize positive impact by focusing on human-centered medicine that AI can't replicate. Use your knowledge of chylomicron and VLDL triglyceride delivery to 1. Educate communities about diet and lifestyle, preventing metabolic disorders in underserved areas where AI access might lag. 2. Advocate for equitable healthcare policies, ensuring AI-driven tools don't widen gaps for vulnerable populations. 3. Combine this biochemistry with your war doctor goal, addressing malnutrition or metabolic stress in conflict zones, where AI might not reach. AI can crunch numbers, but your empathy, cultural insight, and hands-on
but
care.
this knowledge that I'm learning doesn't have to do anything with my empathy.
Fair point. The biochemistry of chylomicron and VLDL triglyceride delivery is technical, not directly tied to empathy. But this knowledge can amplify your impact as a war doctor by 1. Enabling precise diagnosis of metabolic issues like starvation-induced
Anyway, fuck it.
dyslipidemia.
I don't know. Anyway, alright. Triglycerides. Let's talk a bit about triglycerides.
Triglycerides are lipids made of 3 fatty acids bonded to a glycerol backbone, stored in adipose tissue or circulated by chylomicrons from diet and VLDL from liver. They serve as the body's main energy reserve, yielding 9 cores per gram when broken down via lipolysis and beta oxidation, and are critical for insulation and organ protection. Want to focus on their synthesis, breakdown, or clinical significance?
What's the purpose? Is it something to do with storage for metabolic fuel?
Yes, the primary purpose of triglycerides is to store metabolic fuel. They pack energy 9 cores per gram in adipose tissue for later use during fasting, exercise, or starvation, releasing fatty acids via lipolysis for beta oxidation to produce ATP. They also insulate the body and cushion organs. Need more on their storage mechanism or energy release?
And how does acetyl-CoA and the TCA cycle and beta oxidation relate with them?
Triglycerides relate to acetyl-CoA, the TCA cycle, and beta oxidation through their breakdown for energy. During lipolysis, triglycerides in adipose tissue split into glycerol and 3 fatty acids. The fatty acids undergo beta oxidation in mitochondria, where they're sliced into 2 carbon units, forming acetyl-CoA. Acetyl-CoA enters the TCA, citric acid cycle, combining with oxaloacetate to produce energy via ATP, CO2, and electron carriers, NADH, FADH2, which feed the respiratory chain for more ATP. Glycerol can also be converted to pyruvate, then acetyl-CoA, linking to the TCA cycle. This process taps triglyceride stored energy when needed. Want specifics on any step or enzyme?
That's fine. Okay. Let's talk a bit about the hydrophobic nature of triglycerides and what is its significance. Basically, hydrophobic means we need these lipoproteins.
The hydrophobic nature of triglycerides, meaning they repel water due to their nonpolar fatty acid chains, is significant because it makes them insoluble in blood's watery plasma, requiring lipoproteins like chylomicrons and VLDL to transport them. Their hydrophobicity also allows compact, high-energy storage in adipose tissue without water retention, maximizing energy density for metabolic fuel. Want to explore lipoprotein transport or storage?
What are tissue-bound lipases?
Tissue-bound lipases are enzymes anchored to cell surfaces, primarily lipoprotein lipase, LPL, and hepatic lipase, which hydrolysize triglycerides and chylomicrons and VLDL into free fatty acids and glycerol. LPL, found on endothelial cells in muscles and adipose tissue, breaks down circulating triglycerides for energy use or storage, while hepatic lipase, on liver cells, further processes lipoprotein remnants. Need more on their regulation or role in metabolism?
Yeah, and also tell me about the ApoE content, how this relates. ApoB100.
Tissue-bound lipases, like lipoprotein lipase, LPL, on muscle and adipose endothelial cells, hydrolyzed triglycerides in chylomicrons and VLDL, releasing fatty acids for energy or storage. Activated by apolipoprotein C2, ApoC2. Hepatic lipase, on liver cells, processes remnants and HDL, modifying lipids. Apolipoprotein E, ApoE, found on chylomicrons, VLDL, and HDL, binds receptors, like LDL receptors, on liver and peripheral cells, facilitating uptake of lipoprotein remnants after triglyceride hydrolysis.
Can you put everything together in a system that we just mentioned and kind of explain it critically for us?
The system of triglyceride metabolism, tissue-bound lipases, and apolipoproteins, ApoE, ApoB100, ApoC2, works to transport, process,
Okay,
and utilize.
imagine we're a really bored student that hates this, and you want to make us interested in it while remaining factually correct and scientific, yeah?
All right, picture this. Your body's a high-tech energy factory, and triglycerides are its secret fuel stash, like a hidden power bank for when you're sprinting, starving, or just chilling. These greasy molecules are crazy hydrophobic. They hate water so much they can't float around in your blood alone. Enter lipoproteins, the coolest delivery trucks ever. Chylomicrons haul triglycerides from your burger straight from your gut, while VLDLs are like the liver's homemade energy packs. But here's the drama. These trucks need to unload fast, and that's where tissue-bound lipases swoop in like ninja enzymes. Lipoprotein lipase, LPL, stuck on blood vessel walls in your muscles and fat, slices triglycerides into fatty acids, ready to burn for energy or store for later. It only works when apolipoprotein C2, ApoC2, the truck's VIP pass, gives it the green light. Once the truck's half empty, apolipoprotein E, ApoE, acts like a GPS, docking chylomicron remnants or VLDL leftovers to liver cells via receptors, clearing the road. Apolipoprotein B100, ApoB100, the boss of VLDL and LDL, keeps the whole truck stable, ensuring it delivers without crashing. Critically, this system's a masterpiece of evolution. It maximizes energy storage without bulky water, but if you overload it with junk food, it clogs arteries.
Let's talk a bit about the receptors of like LDL, HDL. What is this PCSK9? And also remember, gamify it, make it interesting, make it as fun as interesting while keeping it accurate like you just did.
Yo, let's dive into the epic battlefield of cholesterol transport, where LDL and HDL receptors are like rival fortresses, and PCSK9 is the sneaky saboteur shaking things up. Picture your body as a medieval kingdom, with cholesterol as the gold needed to build castle walls, cell membranes, and forge royal decrees, hormones. LDL receptors, stationed on cell surfaces like liver and muscle, are the loyal gatekeepers. They grab LDL trucks, packed with cholesterol and ApoB100, via ApoB100's key, hauling cholesterol inside for use, then kick the empty truck out to keep the kingdom's road.
Sorry guys, my housemate knocked the door. How unprofessional of me. But, Grog, could you repeat the whole story again?
No worries, housemaid interruptions happen. Let's replay the cholesterol saga, keeping it fun, gamified, and scientifically tight. Your body's a medieval kingdom. Cholesterol's the gold for building castle walls, cell membranes, and royal scrolls. Hormones. LDL receptors are the trusty gatekeepers on cells like liver and muscle, wielding hooks to snag LDL trucks, jammed with cholesterol and ApoB100. ApoB100's like a key card, letting receptors yank LDL in, unload cholesterol for use, and boot the empty truck to keep the kingdom's roads clear. HDL receptors, mostly SRB1 on liver cells, are the eco-warriors at recycling hubs, latching onto HDL trucks loaded with Apo AI to collect excess cholesterol from far-off tissues and ship it back for cleanup or reuse, protecting the kingdom's arteries from plaque invasions.
All right, let's talk a bit about okay, can you do the same thing for fatty acid metabolism to explain beta oxidation and fatty acid synthesis?
All right, strap in for a wild ride through the kingdom of fatty acid metabolism, where beta oxidation and fatty acid synthesis are rival guilds battling for energy control in a biochemical RPG. Picture your body as a bustling medieval city, with fatty acids as barrels of oil, energy-packed but tough to handle. First up, beta oxidation, the warrior guild in the mitochondria, the city's forges. When the city's low on fuel, like during fasting or a marathon, triglycerides and adipose stores get chopped by lipolysis into fatty acids. These barrels need to sneak into the forges, but they're too bulky alone. The carnitine shuttle, like a rogue smuggler, swaps fatty acids for carnitine, slipping them through the mitochondrial gates as acyl carnitine. Inside, beta oxidation's blades, enzymes, slice the fatty acid chain 2 carbons at a time, forging acetyl-CoA like gold coins, plus NADH and FADH2, energy potions for the city's power plant, respiratory chain. Each cut powers the kingdom, turning fat into ATP to keep you running. Now, flip to fatty acid synthesis, the alchemist guild in the cytoplasm, the city's workshops. When you're flush with food, carbs or proteins, the city's got extra acetyl-CoA coins from sugar breakdown, but they're stuck in the forges. The citrate malate shuttle, a crafty merchant, smuggles acetyl-CoA out as citrate, splitting it back to acetyl-CoA in the workshops. Here, acetyl-CoA carboxylase crafts malonyl-CoA, a 3-carbon gem, and fatty acid synthesis, the master smith, hammers acetyl-CoA and malonyl-CoA together, adding 2 carbons per swing to build a long fatty acid chain, like palmitic acid, stored as triglyceride barrels for lean times.
Beta oxidation. Guys, I don't understand anything personally. I hope this podcast at least helped you guys, because I don't understand these things. Yeah, and I got things out of it. I'm not to kind of say I didn't. Thank you for watching, guys. I hope you have a wonderful day, and peace.